Good morning, everyone. Please find your seats. All right, very prompt. Thank you so much. It is my pleasure to welcome you to the final day of GMI15. I hope that you all took a lot of memorable photos yesterday, messaged your family and friends holding a snake, whatever your experience was yesterday. I hope it was a delightful one. I was partially terrified and excited. So there's a lot of unattractive photos of me circling around, I think, with my mouth gaping open. But I'm really glad everyone got into it. I hope you enjoyed the barbecue, and I'm so glad that the weather stayed out for us, even for just a moment. So now that we're on our final day, we do have a 3/4 day. We'll finish at 3 o'clock today. So if we stick to time, hopefully everyone has a bit more of an afternoon to be able to wander around before your flight home. Today's 2 sessions. We'll be kicking off with Dr. Sonia, who'll be chairing session 4 on enabling infrastructure, tools and applications. So I'm going to hand over to her to get us going. Thank you, Dr. Sonia.
Thank you, Tuyet. So now I wish I have a footstool. But so good morning to everyone and welcome to the session 4, the first session this morning. This will be on enabling infrastructure, tools, and applications. So with the global demand for in-house and accessible and analytical capacity increasing, this session would focus on enabling infrastructure, tools, and applications. This session will seek to answer what are some of the tools and use cases of analytical approaches that have been applied to strengthen public health. So for the first speaker, we will have Torsten. Torsten is the lead bioinformatician at the Center for Pathogen Genomics and convener of this event. And to talk about what is the future of bioinformatics, let's welcome Torsten. Torsten.
Thanks, Sonia. All right, so you're thinking, well, why is the convener giving a talk? And well, who is this arrogant bastard that's like giving himself a opening talk? And to be honest, when I was convening GMI, I thought this is going to be awesome. Conveners don't have to give talks. That would just be embarrassing. So I think I tried to delete myself from the program, but the steering committee kept putting me back in. So here I am doing another talk. And I've been asked to talk on the future of bioinformatics. So what is bioinformatics? Like, for a lot of people in our field, it is a bit of a mystery box. Like, they don't really know what goes on in that black box, what goes on behind the scenes. And for some people, it's a bit like the dark arts. You know, we're like magicians or wizards, and we can manipulate this data and do something useful with it. I like to perpetuate that myth. It keeps us employed. A lot of people think our job's easy. There's another whole group of people on the other side who think our job is just pressing a button, you know. We're just sitting around, you know, reading Twitter all day and not really doing much and getting paid for just doing basic stuff that an AI might be able to do. But really, what the first 10 years of genomics bioinformatics was essentially this. It was taking FASTQ files and Excel files and making a report out of them or some kind of summary. And that's what our job has been doing, what we've had to do in our jobs the last sort of since the beginning. But we're now reaching this point, you know, where this stuff is becoming more standardized, a bit more routine, and we're getting asked to answer much more complicated questions with much more types of data. So not just genomic data anymore. We're asked to mix. We want to mix in other data sources and sort of, you know, everything we know about our particular microbe, its virulence, host factors. Like bioinformaticians are, although our original job is now kind of solved, there's lots of harder questions that are going to keep coming, and we're going to need better and more diverse bioinformaticians now, actually. Sorry, this is a new talk. So I guess what I'm saying is that bioinformatics is a new discipline. You know, we've had this model of, before genomics came along, we had this model of small amounts of data, a bit of capillary sequencing, lots of assays. You know, you could fit all your data in a red lab book, a notebook. But now, you know, we're in a new era, biology 2.0, where we're getting lots of data. It's all very omics focused, and we need high performance computing. We need to store it, to analyze it, to make sense of it. And I guess, there's this new career that, has become established now, this new type of scientist. And this new type of scientist, which we're talking about today, is what some of us call it a bioinformatician. Some people prefer to be called a computational biologist. Others prefer to, there might be biostatisticians which fall into this, or I guess the new term that's kind of encompassing all these professions is sort of the data scientist. And lots of my bioinformatician friends who got sick of the industry just went to LinkedIn and changed their experience from bioinformatician to data scientists, right? So we are data scientists, specialised in the biological data. So this is sort of this new occupation that I think is going to become an essential team member of every team. And I guess it's still a vague. Like most of the bioinformaticians here are sort of multi-skilled. We're not just doing one type of thing. It's not like just focus on one project. Some of us have to be computer administrators. Some of us have to be database engineers. Some of us are doing multiple of these things. A new kind of role, like Jake in our lab has become sort of our data steward. This is a new role. We are generating so much data. Somebody has to keep it organised. Somebody has to make sure what we upload to Genome Tracker is good, is valid, and we're not uploading stuff we shouldn't be. Like there's this whole new roles that I think are key parts of a public health lab or even a large research group. What else did I have on there? Sorry. Somebody want to yell one out? A trainer, yeah. So obviously, as bioinformaticians, as you know, we are endlessly called on to train, maybe new students or new lab members or go to educational events because people want to learn about bioinformatics, and we most of us were sort of self-taught, or maybe some of us have been trained. But training, like this is a really multi-skilled occupation. It's probably true of other science roles as well, but bioinformatics has been particularly rapidly evolving that the training seems to be a constant demand. You can't just teach 1 year and then it's fine for the next 10 years. But where do bioinformaticians come from? The stork doesn't bring them in a basket, unfortunately. I guess traditionally they've come from lots of places. So I guess the most common one that happens is that they come from within the existing institute or within the lab, often from the wet lab. These are wet lab biologists who kind of getting bored of sitting at the bench, and but kind of out of necessity as well. Like the lab has got some sequencing data or some other high throughput data, and there is no one there to analyse it. And I guess that's kind of how I started. Like, this new genome Illumina instrument came to our university, and they did a run, and then they realised, what do you do with these 2 files that it produced? And that's how my career started, just Googling how to figure out what to do with these 2 files. And I guess that's where they come. I guess there are a lot of formal training programs in bioinformatics. There's Masters of Bioinformatics and diplomas and people doing PhD in bioinformatics. So that's another common source of students. I find that the formal training is quite mixed around the world, and even within Australia, the quality of that education. Really, I think bioinformatics works better as a sort of a master and apprentice type situation. Like I think the hands-on training is where the reality is. You learn all the real useful stuff sort of in a lab environment with other people. So the lone bioinformatician, which is a common problem in our field, is not a good thing generally. But with online communities that we've established, I think the lone bioinformatician is not as lonely as they used to be, but I still think having someone to bounce ideas off in your lab, 2 bioinformaticians is worth more than double 1 bioinformatician. That's how I think, because they can help each other. Other places is others. In Melbourne, a lot of bioinformaticians come from outside both of those. They're other scientists like physicists and astronomers, where there are no jobs in this country for those people. They go to uni, love it, but there are no jobs for them. So they migrate to bioinformatics because we're looking for people that have that type of thinking. They've worked with big data and maybe done some software engineering or programming to analyse their data in the past. So that's sort of a very common, I don't know if that's true in other parts of the world, but it's very common here in Australia. Oh, why is that font small? Here I'm just sort of, what type of people, what do you need, what are sort of the characteristics of a good bioinformatician that you're going to need to have in your lab? I guess the key one is this logical thinking. So you may think, well, we're all scientists or we're all kind of logical thinkers, but I think, computing and bioinformatics, it's next level. I guess you have to appreciate that when there's a piece of software or a script or a pipeline, you can't sort of skim over steps. Like every line in that script does something and affects the rest of the script or rest of the pipeline. So you really need someone that has that attention to detail and is willing to hold all that information, that flow in their head at once. You can't sort of just gloss over bits and pieces. Like it's not okay that there's an extra microliter of buffer or something. Like that would ruin everything. I don't know. Obviously the lab's strict as well, but I don't know, I just feel like it's a bit more flex. Computers are pretty pedantic. Sorry. Doing bioinformatics. Yes, I'm trying to give a bit of background here before I give my future predictions of bioinformatics. So I guess in bioinformatics, how do we do bioinformatics? Like if you're writing a document, everyone knows you just use Microsoft Word or Google Docs or whatever. It's kind of the same. You do it the same way. But when it comes to bioinformatics, sort of two main approaches. There's this command line interface, which is sort of this Unix terminal. You may have walked into somebody's office and just seen the black background and all the text flying around and keyboards typing. That's sort of what we call the command line interface. And that's kind of what you have to do traditionally if you want to do a lot of high throughput, bespoke analysis of data, big data analysis. But then there's also, the graphical user interface, which you're familiar with. There's lots of options there where you sort of have a graphical way of doing it. So I guess I sort of see it as one is the keyboard method and one is the mouse method. And the keyboard method gives you that high throughputness and the mouse method gives you that sort of low to medium throughput. Although that's changing. A lot of systems are coming along that are trying to take the power of the command line and put it into a graphical user interface. Now, what sort of things are out there? There's most people here have heard of Galaxy, I assume. Galaxy is a graphical user interface where you can sort of upload your data and compose workflows, and it's been around for a long time. It's well supported, and it works great on that low to medium throughput mode. But one of its challenges is, although you can install it on your own computer, it's not often done that. It's usually a cloud service, and it is well funded, so there's nodes in Europe, Australia, and the US. And the biggest problem there is that a lot of countries we discover, our partners, cannot upload their data to it. It's against their data agreement with their government, so they can't export data out their country, so they can't sort of use those things. Terra is another platform that is used in PulseNet. That's another cloud system that has a whole lot of well-defined, actually very good pipelines. The pipelines in Terra are great, but also this exporting of your data problem. There's some sort of commercial tools. So these are just like applications like Microsoft Word or Microsoft Excel. There's Genius and CLC, and they're great for sort of small analyses. I think once you start to do bigger analyses, not so good, and maybe not as focused on public health, more focused on sort of general genomics, not just bacterial. And then we come to sort of more dedicated platforms. So I encountered, has anyone heard of EpiSeq, the bioMérieux tool? Yeah, so it's aimed mainly, they're targeting mainly hospitals and clinical pathology centres. So it's actually very good. Like, it is a graphical user interface. It does what you want. You can just give it hundreds of FASTQ files and select them and upload your spreadsheet and have metadata and build a tree, build a cluster. And I was actually a bit skeptical of it, but I sort of had a look at it and it was pretty good. And then I wondered how are they doing all the analysis? And I spoke to the salesperson. He wasn't quite sure, and he dug down and finally took me to this about page. And it was all a standard Unix. Half of them were my tools. Half of them, all the tools that, I said, I wrote that. And he, oh, okay, don't think he believed me, but it was a good sign that these tools, even the commercials are sort of integrating them into their products. And BugSeq is another option. There's lots of these new, I don't know, BugSeq comes out of Canada, I'm not sure. And there's many of these platforms, cloud platforms, that all do the similar thing. You upload your FASTQs, you upload a spreadsheet, they build trees, the standard thing. And I think they are a potential good option for anyone that can't sort of have their own in-house solutions. Obviously, you pay for these and you have to be able to upload your data, but they're actually all using the same underlying tools underneath. So, and I guess I should have put this slide beforehand, but just where are we doing our bioinformatics? when we go to some LMICs, they're doing their bioinformatics on a workstation or a laptop in their lab. It's not even like a shared, not even necessarily got a shared drive connected to it. Others are doing it, you know, they have their own high-performance computing, or they have one at their institute, and of course there's the cloud. And strangely now, there's actually a lot of, everyone is pushing to the cloud, but there's been a bit of a regress back now to having what we call on-prem, so having your own hardware. And the reason for this is sometimes to do with government issues, exporting your data out, but also the cloud. The dream of the cloud is kind of good, but it's also very expensive for a lot of people. And if you don't have someone in-house that can help manage the use of the cloud and, you know, minimising its resource use and tweaking its resource use, you're going to end up with a massive bill. And yeah, at our lab, we've sort of used on-prem for various reasons, and yeah, probably saved millions of dollars over the years. But you do still need that, you know, that bioinformatician that can also understand cloud and DevOps and things like that. The other problem is operating systems. It used to be a problem. You'd have these different operating systems and software wouldn't run on one or the other. But when it comes to bioinformatics, it's kind of been solved over the last few years. So Macintosh is a Unix type operating system. Linux is obviously a Unix type operating system. And Windows in the last 8 years or so has added a layer called WSL, which Windows Subsystem for Linux. So basically every Windows installation these days has Ubuntu Linux installed on it, and you can like boot it up, and you can essentially have a Linux, a Unix computer built into your operating system. And what this has meant is that these three major platforms now are all kind of running on the same underlying, can run on the same engine, this Unix compatibility layer. And once you're compatible with Unix, you have, the whole world opens up to you. And the world has converged on Bioconda. So Bioconda is a packaging system for software, for bioinformatics software in particular, life sciences software. And yeah, most of the tools we all use now are in Bioconda. They're regularly updated. There's a sort of a big community of maintainers in this project, and it really makes it easy to install. Because as many of the bioinformaticians here, or for anyone that's had to install any bioinformatics software, you know how much time it took to install stuff. And now that's kind of been solved for us, and we can focus on the more important stuff, how to combine these tools together rather than just getting them working. So how does this all fit into the future? So I guess I wasn't sure what to put in this talk, so I thought I'd just come up with a few things that I think we're going to be facing in the coming years. So I'm going to look into my crystal ball. And the first thing is something that I predicted like 8 years ago and was wrong. I thought once nanopore sequencing came along and that we would suddenly just be doing everything with long reads and generating closed genomes. Instead of these draft assembly genomes in 150 contigs, we would have complete replicons, plasmids and chromosomes, and that would be our basic unit of exchange. But that did not happen, and I think it will happen. I still predict it's going to happen, but my dream is that we no longer FASTQ files are not handed around as a primary source. Like, it's ridiculous. It's like, you know, 0.5 gigabyte just to store a small genome. Like, I think the genome will become the single unit of currency in the next 5 years, hopefully. But not as you think it. It's not a FASTA file with 3 contigs, because that is not rich enough to represent the actual content of those FASTQ files. We need to store the genome graph, because genome assembly generates a mathematical graph, and we throw that away when we generate this simple FASTA file, which is a standard from the 1980s. So I think it's going to be a richer format, which is going to be small. Instead of 500 meg, we're going to have 5 meg. It's going to be the assembly graph, and then if there was any diversity within that sample, the graph will encode it, or there'll be some side information included with it to show some of the variation. And that will be important when we start handing around assemblies of metagenomes and wastewater samples and these sort of more diverse samples. But I do think it will happen, and FASTQ files will always be there as sort of the primary data, but we won't be handing them around. The next big thing I'm thinking is going to happen is, I just talked about Conda and all these things, but that still requires quite a bit of expertise. And I think what's happening now is this bioinformatics in the browser is going to sort of take off. And I'm partially invested in this idea. So what this is, so there's this thing called WebAssembly. So what people have done is they've added another layer on top. They know everybody's got a web browser. And WebAssembly is essentially kind of like a virtual computer that runs inside your web browser. And it's not specific to Chrome. It does run on all the web browsers. And it's really a whole new virtual computer that you can write software for that will run anywhere as long as you have a web browser. And it's more powerful than just say JavaScript, which runs in every web browser. JavaScript's a sort of a slow interpreted language. WebAssembly lets you kind of get very close to the hardware. So if your desktop computer has a fancy GPU, or if it has some kind of special hardware, that is directly available to the WebAssembly. And now you can compile software to this WebAssembly target. Instead of compiling it for Windows or for Linux, you can compile it to WebAssembly and it will run anywhere. And it's starting to really take off. So the R statistical language programming language, they're heavily invested in writing all their code to run on WebAssembly, and lots of other big projects are now targeting WebAssembly. So I think that is the big future. I think we'll have, this means that you can do your bioinformatics in your browser. Labs with no experience will be able to just sort of go to a web page, pull down the applications, and they can run all the standard command line tools or platforms in their web browser at nearly native speed. The other unfortunate thing that I think is going to happen is that we are going to continue to be siloing ourselves. We saw here that everybody's writing their own platform or looking to write their own platform, and I think that's probably a necessity just because of how governments work. And they're all very similar, but they are different. But I think what's going to become important is that they talk to each other. And this is this whole idea of interoperability. And I guess what I think is going to happen is that all these platforms, say AusTracker and SPS, Swiss Pathogen Surveillance, SPSP, working together, if we might, I've just demonstrated sort of a one-to-one here, but this would be one-to-many. Say I have my database of what I have and I notice some pattern. So instead of, I'm not allowed to share my whole FASTQ files or my whole assembly for whatever reasons, but what I might be able to share, because that's too specific, it tells you every snip, every detail, what I might be able to say is, what I should say is there's this technology called sketches, or you could think of it as CGMLST profiles, but there's a way of reducing a genome down to a smaller amount of data that is hopefully high enough resolution to be useful. So you can send this sketch, and you can't recover the genome from this sketch. You could say, here's a sketch, does anyone have anything like it? And all the other platforms also have their database, their library of sketches. And the thing about sketches is that they're very fast to find the distance between them. So they easily rule things out, but they're not guaranteeing to rule in, but they can find matches quickly, like very quickly. Like you can compare millions of sketches very quickly. So you can find potential matches. And then we do what we humans are actually good at doing, which is when you find a match, you actually talk to each other. So that we need to set up a system there, whether it's talking, phone calls, emails, whatever, some kind of alert. And then you start the conversation and you say, look, yeah, you're right, we do have some ST5s with this virulence gene. Maybe we can share. And then my hope is that then there'll be some bilateral agreement where you share the actual FASTQ if you want with each other, and then move on and do the analysis in detail. And I think this process, if you do it slowly over time, people build trust in this system, that they haven't been burnt, and then they'll start, why should I just share it just bilateral? Let's just share it with everyone in the network, the global network. And I think over time, people, once they build that trust, they will do it. Another big thing that's going to happen is genotype-phenotype. I'm not going to talk about it. We have two speakers here, two young, handsome speakers who will be talking about that at the end of the session. And I guess what's always happens in our field, technology changes. DNA is just the bottom of the pile here. There's lots of other biological artifacts and molecules that we can sequence and use to assay that makes things cheaper and faster. So bioinformaticians are always going to have to adapt to new technologies, new approaches. DNA is not the end of the story. And one day, I predict that Marc's vision will come true. There will be a united genome tracker where everybody does share their data quite freely, and there's all sorts of magic tech to make it not get stolen and keep everybody happy. That's what I hope to see one day. And finally, I hope to see some kind of formal joining of the three, the trinity of conferences that we're sort of working within, as Tiet said earlier in the breakfast today. I hope PHAGE, IPCN and GMI will become a bit more coordinated, maybe have cross-fertilised working groups and so on. That's what I think the future is. Thank you.
Maybe we can entertain one question. There you go. Sorry, Torsten.
Who's asking you a question?
Who's first? We got two, sorry, Dr.
Okay. Torsten, yeah, the one thing that's causing challenges for me at the moment is, how do we share the stuff that comes out of metagenomic sampling? So, because the notion of a GFA is still basically thinking about an isolate. So we get an isolate, and then, we're interested in this. But like, especially with wastewater and environmental sampling, we're getting a kind of snapshot of a whole other things. So then we kind of stuck sharing the FASTQs, and that's really not great because we might not want to share everything in the FASTQ. It really it's too big. It gives us all sorts of headaches. So yeah, what do you think about that?
There's nothing stopping you from putting a metagenome in this sort of format. What do you think is stopping you? Why can't it be used? Because it's a mess, or
like so basically GFA that comes out of Metaspades, that kind of thing. Okay.
Yeah, I think it can still work. It depends on what you want to use it for. I think people are stuck at metagenomics because it's we don't get good results out of it. Like it's 1 billion contigs, and people try to bin them into species groups, mags, and that's not very successful. But we're just not doing deep enough, good enough long-read sequencing. I think that can untangle a large amount of the metagenomic mess that we have. But yeah, wastewater is a bit different. Maybe it's very sparse, but then the graph format still works then as well. It may not be a graph format, Peter. It may be another format. But I think there's a lot of... The information content in a massive FASTQ file is not that high compared to how much space it takes up. Maybe that... I think we need something simpler that we can compare quicker, process quicker. But yeah, a fast A file of contigs is not good enough at the moment, I think. Yeah, but it's a good question. I haven't fully answered it. I get to gloss over it a little bit.
Just one more for you. So when Genome Tracker was during the first inception, we said, put your data into NCBI, and now with over 2 million sequences, we're saying no one can really scoop you, that kind of stuff. Do you think, or have you had conversations with NCBI about a section for sketches?
Yes.
So that then maybe it's kind of a baby step for people to put stuff into NCBI?
Well, I tried to have... I've spoke to Bill and Klimke and others about this idea before, the sketch idea, and he's aware of it. At first I was thinking more of it, NCBI should be sketching everything and giving us an API or a way to query our genomes against the database. Because the common problem now, people want a reference genome, or they're looking for something similar to what they have for contextual data for their phylogenetics or phylogenomics analysis. And how do you do that? I think how everyone I know does it, they go to GenBank and they download every species. Like if they're working on Acinetobacter baumannii, they download every Acinetobacter baumannii and then manually compare. And everybody's doing that all the time. It's ridiculous. They could just send a sketch or a CGMLST, some kind of small profile that NCBI can rapidly match against everything they have and tell us what matches.
Yeah, the modern blast.
Modern blast, yeah. I mean, blast, yeah, exactly.
It's a little different, but
yeah, so...
We've discussed this, and I think we can put it on the table. Happy to do so.
Yeah, I mean...
GMI could put it on the table if we had consensus.
Yeah. I mean, obviously NCBI needs the resources to do this, and as you know, I've had a campaign for 10 years to get MLST into pathogen detection. But yeah, NCBI is under-resourced, and yeah. But let's do it. Let's march onto the campus and march on the pathogen detection building. All right, thank you.
Thank you, Torsten. So the next presenter is Dr. Wytamma Wirth. He is a technical biologist based at the Peter Doherty Institute, where he works on different aspects of genomic epidemiology, phylogenomics, and bioinformatics. So Dr. Wytamma will talk about building the Center for Pathogen Genomics Bioinformatics Analysis Platform. Dr. Wytamma.
Morning, everyone. Thanks for letting me present this work. I actually changed my talk slightly, so I'm just going to give a demo sort of the platform that we've been building. I'm very happy to present on the Center for Pathogen Genomics Bioinformatics Analysis portal that we've been developing. And so this is just a screenshot of what the portal looks like. So basically the idea is it's a job running portal that gives you a graphical user interface to lots of different bioinformatics tools. So I think something that, like Torsten was just saying before, there's a lot of these platforms out there, and this platform was sort of proposed as a way for us internally to give access to our tools and to help some of the countries that we're working with access those tools easily, and to really lower the barrier to running these analyses. And so the idea is sort of for the platform to sit somewhere in between this sort of EMBI job dispatcher and Galaxy. So having a little bit more feature rich than the job dispatcher, which is sort of just these web forms to run analyses, and then not as complex as Galaxy, which has is very feature rich and has a lot of workflow builders and things like this. So we're sort of trying to sit just in the middle between these two. I just have like a little screen capture. Will that work? Go.
Is it a video?
Oh yeah, here we go. Yep. So yeah, there's just logging in. So you can see that there's a bunch of different tools on here. So just going to go to MLST and then grab a genome that I have, and you can sort of drag drop it to upload, give the run a name, and then submit that job. So it's very quick to get an analysis going. We're just drag and dropping to run. There's a lot of this sort of configuration and stuff like that. And then once you get the output, it does some output rendering. So for like CSV files, JSON files, we're like building out different types of renderers for like images and trees. We have some experimental features built into it. So here I'm like demonstrating some AI features that are summarizing the sort of MLST report that we get. So we get this summarization of a report saying that, okay, this is Salmonella enterica ST19, and yeah, give some follow-up steps and some summarization of that. There's some like search features built into this. So you can go through and find some Salmonella typing and yeah, sort of do the same thing, reusing that file that we just uploaded to run it. We get like real-time logs of the results as the tool is running. So for like longer running jobs, you can sort of monitor that. And I might just skip forward for this next part. So yeah, there's a JSON spec that we have that configures these tool interfaces. So basically this whole interface is dynamically generated given a specification file. And so yeah, enables you to add different types of like drop-down lists to select things, or different input validation. That's all sort of handled just through this specification. And we can do things like tagging runs and renaming things and uploading files. There's sort of a runs management section. So very basic, but enables you to go through, see old runs that you've started, stop runs, delete them. This is you saw before, the sort of output page. So this is for Abratamer, an AMR tool, gene prediction tool that we have developed at MDU. And yeah, so you can see here that you can see all the file outputs from this running this tool, and then this sort of results section where the table that's generated is rendered there, and we can sort of tab through the different results types. Also in that page, we saw before, there's those real-time logs there so you can monitor stuff as it's running. And then it has some sort of more, extra details there. So it'll show you the exact command that was run, as well as the environment that it was run in. So it sort of enables this reproducibility aspect. And we have run sharing built in. So you're able to enable for a particular run or set of results. You can say, okay, I want to share this result with someone else and configure that and send someone the link, and then they can access the results from a particular analysis that you've performed on the portal. So it has this basic sort of file management system, which is all configurable to like spaces and file uploads amounts and how large files can be uploaded. So it's very basic. You can go through the files that you've uploaded previously, delete or download them, and save the outputs from previous runs into like your sort of file management system. One thing Torsten was speaking about was WebAssembly. So we actually have this WebAssembly version of Jupyter notebooks built into the platform. And so basically this is that the use case for this is that you have sensitive data or local data that you don't want to upload to an instance of this platform. So you can actually do your analysis in your browser by downloading. It has a plugin, so you can download samples from your file set. So once you can click on the secure analysis environment link in the portal, and that will show you this list of files that you have. That's that same file list from before. And then you can just press download, and that will save it into your browser. And then you can run code in your browser on that data. And so if you had like your samples or assemblies or something like that, or a tree, and then you wanted to put patient data visualization on that tree, you can do that by just adding that into your browser, and it will never be uploaded anywhere. It all stays on your computer, and you can run your analysis in this sort of curated environment that's local, but has access to your files on the portal. So we've been experimenting with some different sort of AI integrations in this. And so per tool, you can sort of configure AI integration into this and develop this sort of report interpretation or this, you can see there's a little red warning up the top there. So you always have to be careful with this sort of stuff, but from the sort of testing that we've been doing, it seems to work fairly well for this summarization and follow-up step recommendation template that we're using currently. Jane might have saw that when I just searched for salmonella before, AMR rules appeared. And if you remember on Monday, Jane presented on this tool, AMR rules that they're using. And so during her talk, I was able to add that tool into the platform. So I just wanted to show how easy it is to add this. And this is through another integration with large language models. So we've developed a CPG tool building GPT instance through ChatGPT, but we could sort of apply this to other LLM platforms. And so this particular tool builder is given a GitHub URL, and then from that GitHub URL, it will generate the spec that's required to run that tool. So this spec, contains all the sort of things like the version number, URL, different licensing, which it develops the tags, it writes the actual interface, so the parameters that are required as inputs, and the sort of command line command that you have. It's dependent on the quality of the GitHub repo that you give it as to like how much information it can actually extract. So if you have good docs, this works really well. If you don't have good docs, it gets maybe 80 or 90% of the way there, and then you can kind of tweak the spec a little bit to generate it. But so I was able to put in that, put in the GitHub URL to AMR rules, and then this, and it generates this interface, which then you can run, and it will and works. It has it generates a Conda environment as well, so that it can install it. I had to tweak it just a little bit. Maybe it was like 98% of the way there. But yeah, so very quick to be able to generate these sort of graphical interfaces to CLI tools. Has dark mode. It's just just a plug. It's also designed to work on mobile. So, just as easily you can run this on your mobile phone. If you're out and about and for whatever reason need to do some sort of genome assembly, you can just run that on your phone. I had a talk about being at the pub and getting emails from your boss and having to quickly run an analysis and send it back to them. So it works really well on your phone. It's all open source and MIT licensed. So it's a containerized platform. So it uses Docker. Assuming you have Docker installed, it's very easy to get it up and running. You just clone the repo, go into the directory of the repo, and run Docker Compose up, and that should start the whole instance, which is then configured with this environment file. So you can configure your, different whatever configuration you want to add to make it more specific to your particular instance. And the GitHub link is there in the QR code. I think I'm going very fast because I haven't heard the bell, but I'm kind of towards the end. Oh, did it? Oh, I just didn't hear. Cool. So there you go. I'm at the end. I wanted to say thank you to the Portal beta testers. So we had a bunch of different people try it out, and especially thanks to Torsten and Louise Judd and Amar Aziz, who have been really helping a lot with the development of this. We have a main instance, so portal.cpg.unimel.edu.au, where we are hosting this instance, but it's not really designed for us to host. It's sort of like, run an instance of it yourself. I think that's kind of all I wanted to say. Thank you very much.
So we'd like to take one or two questions.
Thanks, Wytamma. It's really, really cool work. I was just wondering, how hard do you think it's to adapt that GPT instance that you have to sort of standardize things for other applications? Or is it like a very sort of purpose-built?
It's like it's really just the pre-prompt. It's through OpenAI's platform, you can create these like instances of GPTs, and it really just allows you to add a pre-prompt that you can then share with other people, and it lets them run it. So really, it's not like trained to like do this. It's just the custom pre-prompt, which we've sort of figured out works reasonably well.
It's easy on the resources as well. Like it's not you don't need a lot of infrastructure to run it.
To the LLM or the
because
platform?
It's already pre-trained and
So that one is just running through OpenAI. So it's just you just paste your link in and then it will generate that using their infrastructure. But it's really just the config that it's generating. Then you take that and put it into the platform.
Great, thanks.
Very very interesting tools. Just wondering if this tool also the connection with the SARM high performance clustering is what is inside, or just a connector for the APC configuration?
So like the job runner itself. So it's just using a simple job running batching tool. So it's configurable, so you can sort of limit the number of jobs that run at the same time or the number of jobs a user can run, this kind of stuff. But it's quite simple. It's not designed to like go off and run Amazon batch or anything like that.
The second question is, I saw a single sample analysis. I think you also have the possibility to configure multi-sample analysis.
Yes.
And what about adding also metadata because at the end they want to go to tree with the different metadata. It's typical. How do you do that?
Well, the idea, the portal just sort of treats all tools as like a black box, and it will just run them. So I would put that onto the tool developer and say, you have to make a tool that will do that, and then we will generate the interface for that and run it, however that is run. Thanks.
Thank you, Dr. Wytamma, for the great presentation. And good discussion. So the next presenter is Dr. Sandy Mak. She is based at A-Star Bioinformatics Institute in Singapore. She supports regional capacity building through collaboration with GSAID, the Asia Pathogen Genomics Initiative, and the NUS Center for Infectious Disease Emergency Response. To talk on GSAID or viral bioinformatics, let's welcome Dr. Sandy Mak.
Thank you.
So thank you, Dr. Ma. We're running a little bit late in the time, but I think the discussion is all worth it, so we ask for your forbearance on this one. So for the next presenter, it would be Dr. Ebenezer Foster-Nyarko. He is a postdoctoral research fellow at the London School of Hygiene and Tropical Medicine of the United Kingdom, where he applies computational genomics to uncover the genetic drivers of AMR and virulence in Klebsiella pneumoniae and E. coli. He will share his work with us today on the development of Kleberate. Dr. Ebenezer.
Good morning again, and I introduced myself yesterday. Today I'm just presenting on a tool developed by the Holt Lab. So Kleberate, initially published in 2021, the paper is here. I've put the barcode so that, I still say barcode when I mean QR. So you can have a look. Initially was designed for Klebsiella species complex as a framework and a surveillance tool. And it provides data on what I've listed here. So assembly QCs, speciation, MLST, virulence, and AMR, and then serotyping. So you can see here the tree of Klebsiella, the Klebsiella species, pneumo species complex is shown here, Oxytoca species complex as well, and recently we've added in modules for E. coli, which I will speak about in a bit. So Kleberate takes in as input a genome assembly, be it Illumina or long reads, and then it will output a text file which will summarize the data. And the new version incorporates Minimap 2 instead of BLAST, which makes it super fast. So within a few seconds, you get a report for a single genome. So the structure as well is modular, which means that you can add, different modules, and the different modules are run in parallel, and so if there's a bug, you can easily fix that, and also you can keep adding for different organisms as you need. You can specify with the -P here for either KPSC or E. coli or K-Oxytoca. So this is the way it runs. The input assembly would then go through the general modules, which will output for you your assembly stats, and then you have MLST and then the virulence typing. I'll talk about these first two. This is, I'm preaching to the choir. I think we're all familiar with MLST. Where there is not an exact match, we will attempt to give you the closest match in terms of the number of locus variants. We have the virulence scores, which tells you, looking at the ICE-KP and plasmid-associated virulence factors, a score between 0 and 3, depending on what your genome has. So, for example, if you have just Yersinia bactin, you have a virulence score of 1, and when you have Aerobactin and Yersinia bactin, then the score jumps up. And so that is a really helpful way of summarizing the pathogenicity of the genome. And then for the AMR and capsular typing, we've included, we use CAD, and then we've recently included a way to predict the ciprofloxacin phenotype based on curation that was done from the CLEBNet GSP consortium using thousands of user submitted data. And so you can see on the right an example of the report, which is now incorporated. So in addition to just having the genotypes predicted, you then can see what the phenotype is likely to be. And the latest module uses outputs in addition the reports in a harmonized format using the phage kind of framework that was produced, and this makes it then interoperable, and you can put that into other tools. Again, we can you get the AMR score, which tells you whether you're having ESBLs only or you're having some carbapenimases as well. And then for the serotyping, this uses captive. Captive is maintained by the YS group over at Monash, and there are new features being added, for example, structural predictions and serological validations, etc. You can read more about this. And recently we've added in a module for E. coli, so we bring all the tools for genotyping E. coli together. You can have your MLST, your serotyping, your phylogroups, groups, sugar typing is happening, AMR as well, and then the pathotype assignment is also included. And because it's modular, it's efficient, it scales to large data sets, and again, we have the harmonization standard, which is quite nice. So we've tested this using the eCore Plus collection available in Enterobase, and we found it to be super accurate. And the application there is that we can then use this for surveillance. For example, in some of the African CDC groups working on Klebsiella, recently we had a workshop where they tested this, and it was super efficient. They were able to, within minutes, generate results for their data. So I think the fact that this brings all the tools together in one pipeline and easily integratable into other existing pipelines makes the tool really helpful. For example, Pathogen Watch uses this. BixDB as well incorporates Collaborate, and so it's a way to support large-scale surveillance, and everyone is using the same kind of tools and getting the same results, so you can compare them, which is something that we've been talking about here, that the results should be able to talk to each other. So that is another key feature of that, which makes it really helpful. And these are actually the guys who have been doing the work. The hardcore coding behind the tool is happening here. And just to thank you for your attention.
Thank you, Dr. Eben. Any questions for Dr. Eben? Oh. Okay. He will still be here for the rest of the day, so we can approach him for any question on Collaborate. Our next presenter is Dr. Andreas Stroehlein. He is a bioinformatician and research scientist specializing in pathogen genomics based at the German Federal Institute for Risk Assessment. He works on the development of microbial genomics tools and pipelines and contributes to routine genomic surveillance program in food safety. To present on inferring phenotypes from genome assemblies, implications for genoserotyping and surveillance of Salmonella, let's welcome Dr. Andreas Stroehlein.
Good morning, everyone, and thank you for the GMI organizers to allow me to speak here today. Today I want to talk a little bit about a tool that we developed which allows us to infer phenotypes from genome assemblies. And I want to talk about it sort of in the context of genoserotyping and surveillance of Salmonella, and in the context of the recent shift towards sequence-based typing. So the German regulations for control of Salmonella defines 5 serovars that are controllable by law if they're detected in poultry. And the European regulations actually still rely on slide agglutination assays and the Kaufmann-White scheme for the detection of or the determination of these serotypes. And so this method
relies on the detection of a specific reaction of a series of antisera that detect the lipopolysaccharide structure on the outer membrane of these bacteria. And that tends to work really well if the bacteria have an intact lipopolysaccharide structure, like you can see on the left here, with a lipid A complex anchored in the outer membrane of the bacteria, then an inner core, an outer core, and a variable number of O antigens at the end of it. But some strains exhibit defects in this structure, and that leads to them not being typable using this method. And because of their rough appearance on the microscopy slide, they're called rough subtypes or rough phenotypes. And you can see a number of different variants of this phenotype here. So a number of different defects to this structure due to a number of defects in different genes that are involved in this LPS assembly. And because LPS is a major determinant of virulence for these bacteria, a defect generally leads to reduced or no virulence at all. But what studies show is that it is not always the case. There are actually some strains that still exhibit virulence and are still able to cause colonization and infection. And so that represents an issue in terms of control measures, because if using that method we're not able to actually determine the serovar, that means we might not be controlling things that should be controlled. But as you will know, Salmonella typing has had a long history, and there has been a lot of development over the last few decades, and particularly in the last 10 years or so. Sequence-based typing has really become sort of the method of choice, and with whole genome sequencing becoming commonplace. In many places, this has replaced the traditional methods, and partly due to the fact that it provides more detailed information, such as the information about AMR genes, virulence factors, and so on. But what these methods don't do, they're not able to distinguish between LPS deficient and LPS intact strains. So this information is lost, and all these strains will be typed exactly the same. They will be assigned the same serovar. And so that creates sort of the reverse issue that now we're basically, if we rely on sequence data alone, we will treat them all the same, no matter if there's an LPS deficiency or not. And this is particularly pertinent in the context of an update to the European regulations, which are coming in place next year in 2026, which will require all member states to do sequencing of Salmonella isolates that are suspected to be implicated in foodborne outbreaks. And so in order to kind of understand a bit better the relevance of this particular phenotype, we did a big study for Germany, selecting more than 12,000 isolates over a period of 5 years. And what we see is that somewhere between 0.5% and 13% of samples contain these LPS rough isolates. And you can see that it varies quite a bit between the individual federal states that were looked at in Germany. And so what was interesting was that some of these isolates that we looked at were also part of foodborne outbreaks, and we also detected some clinical isolates, so further supporting that idea that not all of them are non-virulent. And so even though there's a relatively low number of LPS rough isolates that we found, it has some relevance in the context of food safety and public health. And so because of that, we thought, okay, maybe we can devise a method that allows us to sort of create a relationship between the genotype that we see and the phenotype. So somehow integrate the sequence data that we have together with the serological data and correlate observed genomic combinations of genomic features with the assay readout. And as you can see on the left here, basically we sequence every non-typable isolate using whole genome sequencing, and somewhere between 20 and 80% of isolates that are LPS intact. And so we did that. We developed a method that is able to predict the phenotype from sequence data alone, and also map the genotypic diversity that we see across this phenotype. And so this method is called RUFFY, and the tool RUFFY relies on a so-called decision tree classifier, which is a supervised machine learning method, and that requires a bunch of input data. So as training input, you need genome assemblies, and for each isolate, you need the associated phenotype, and then a number of genes of interest, and in this case, we're using all the genes that are known to be involved or suspected to be involved in LPS biosynthesis, which are 53 genes. And then some additional information around the gene clusters that these genes are encoded in. So those are the WAA and WBA gene clusters. And then so from the genome assemblies, we can then extract a number of genomic features. And the current model extracts 5 features per gene and an additional 6 features per sample. And I've just listed a few here. So those are not all of them. Things like the presence of the gene, the length of the gene, the number of copies of that gene, whether certain regulatory RNA elements are present, the serovar and the serogroup of the sample, and then the gene profile. So basically the presence and order of genes within these gene clusters. And based on this data, you can then create a decision tree classifier, and you can see the final decision tree on the right here from our model. And so basically each path through that tree now represents a combination of these genomic features that leads to a prediction whether the phenotype is rough or smooth. And if you now have an isolate for which you do not know the phenotype, you can simply extract all those gene features or genomic features and follow the path through that tree, and you will end up with a prediction of the phenotype for that isolate. Just a few metrics on the performance of the model. So we trained it on more than 4,000 Illumina assemblies from 46 different serovars. We did a 10-fold cross-validation, so randomly selecting 80% of the training data for training and then predicting on the remaining 20%. And we did that 10 times just to make sure that we're not sort of overfitting based on the training data that we're using. And you can see we achieved a 99% accuracy, 94% sensitivity, around 99% precision, so just a little over 1% false discovery rate. And the final model uses 89 features of a total of 821 features that were extracted from the genomic data. And the top 7 features actually explain more than 60% of the variation. So on the right here, you can see the output of the tool, which is basically just a table that tells you per isolate whether the phenotype is rough or smooth. It also gives you a confidence value, how confident the tool is in that prediction, and it gives you the name of the gene profile, and then another file gives you the actual order and presence and absence of the genes within these gene clusters that we looked at. And what we saw was that there are 292 unique gene profiles and of a variable number of occurrences. And what that really shows is that that phenotype is much, much more diverse based on the underlying genomic data than it is described in the literature. And then so next we want to have a look at all the individual paths through that decision tree that actually predict and explain the rough phenotype. And so we found 51 of those. Many of them were known mechanisms that have been described in the literature before that lead to this LPS defect. But we uncovered a whole range of potentially novel mechanisms with genes involved that haven't been described to be involved in the existing literature. So I guess you can see it's really a really complex phenotype with lots of moving parts, lots of genes involved. But one common theme that we really found was that of insertional deactivation. So we saw a lot of cases where sort of insertion sequences or transposase-mediated mechanisms disrupt genes within those gene clusters. So that seems to be a common mechanism. And then next we want to have a look at whether there are clonal lineages established along particular food production lines in Germany or things like that. And you can see on the left here that wasn't really the case. So each of those circles represents a 20 allele distance CGM-LST cluster. And the darkness of the circles represents the fraction of isolates that are LPS rough within that cluster. And you can see there really isn't any evidence of true clonal lineages or strains that have established over this period of the 5 years. And you can see the same sort of in the tree on the right here. These rough isolates are sort of scattered around the tree and seem to pop up sporadically. And then, yeah, just to summarize, so I hope what we've seen is that Ruffy provides this backwards compatibility to the slide agglutination assay, which is still written in the regulation at this point. We have also tested on a number of different applications, so in the context of exploring genotype-phenotype relationships, and I've seen that it works really quite well for other questions as well. So if you're interested in that, I'm happy to talk to you about it. We haven't seen evidence for clonal spread of these strains among particular food production lines or in wildlife in Germany. And one thing I haven't really talked about, but I think that's also important in this context, is that LPS deficiency is not really fully represented by the CGMLST schemas, so only around 50% of the LPS gene loci are part of the CGMLST schema, so it's not really reflecting that diversity that we see. And I guess taken together, what we want to advocate for is to include those isolates in outbreak investigations. So just doing genoserotyping with the established tools, doing core genome-based cluster analysis, but then sort of doing a specific phenotypic subtyping on top of that, just to get the most accurate readout in an outbreak situation or outbreak context. And yeah, with that, I want to come to the end of my talk. I want to thank all collaborators at the BFR, also the people at the FDA who we work with on this project, and also everyone at the Robert Koch Institute who's involved. Yeah, thank you very much.
Thank you, Dr. Andreas. Questions for? Oh, there.
That was a really great talk. I'm just curious what your thoughts are on the issue with losing some of the phenotype data, since it's so primarily sequence-based now. Do you ever think there will be a time where we will get to a point where we can infer phenotype data from sequence with reliability?
You mean inferring phenotype without having a sort of training set on which we can? Yeah, I mean it's a lot more difficult.
Yes.
Maybe Adrian is going to talk a little bit to that. I don't know. But yeah, I think the reason why we were working on this was mainly sort of to prove that we're not losing that information and that we don't need all the methods anymore if we move towards sequence-based typing. I think from a regulation and policy-making point of view, that sometimes it's necessary to get everyone on board. So that's the main reason.
I understand you're probably using decision trees because they're interpretable and
yes, correct.
You can describe the phenotype and so on, and you're already getting excellent accuracy. I'm just curious whether you tried like random forests.
Yeah, we did. We did random forest. You lose some of that interpretability, and it actually didn't perform as well.
Right.
So we were a little bit worse on the random forests, and so that. But yeah, we really want to use it for in terms of interpretability. So I think that was the reason why we chose that method. Yeah.
Thank you.
Thanks.
Thank you, Dr. Andreas. So the next presenter is Professor Adrian Egli. He is the Chair of Medical Microbiology and Director of the Institute of Medical Microbiology at the University of Zurich, and joins us again for a second talk to talk on prediction of AMR from genomes. Are we there yet? Let's welcome Professor Adrian.
Thank you very much for the kind introduction and again the opportunity to talk. This is clearly a topic which is perhaps a bit more debated, and I give you now my personal opinion on this. So when you have such a question, the obvious thing, what you would nowadays do is ask ChatGPT. So thank you very much for the opportunity. I close the talk. So ChatGPT is pretty clear. It says no. Now, is this hallucination? Let's try to look into the evidence. So the first thing I think which is very, very important is to realize what is your use case, because it really depends on your use case, what kind of resolution you need. And maybe in some cases, a simple prediction, absence and presence of a certain gene might be sufficient information to draw some conclusions. So especially if we talk about wastewater surveillance or food surveillance, there you clearly do not need MIC resolution because you're not treating a patient, and for this reason it might be easier. So these would be the lower two examples. But if you go into the clinical situation, then obviously MIC prediction would be very important, and that's the holy grail of bioinformatics at the moment, where you really, you depend, you have a critical ill patient, and inferring then from a genotype an exact MIC, and that would then steer the dosage of a patient. That's a completely different game. There are examples in this realm where I think it already works. So TB, for example, is a very good one, where the mutational space was mapped to a large extent, and there's lots of data around. For example, the Cryptic Consortium I put here as an example. And very often in the clinical setup, we have these kind of curiosity-driven things where we see a certain phenotype. So, for example, it indicates there's a carbapenemase, but our PCRs are negative, so obviously it could be something new or something different. And then you're probably in between these two. So, but let's go back to the phenotype and really start with the basics. And I think there's clearly a reference method for phenotypic antibiotic susceptibility testing. This is the agar plate dilution. And you can see this here on the top. What you do is you have different agars with different concentrations of the antibiotic. You drop your bacteria on it, and then you compare to some baseline, how well do these bacteria grow. This is extremely annoying to do, very manual. It takes ages, and clearly it's not done in routine diagnostic labs. This is something which reference labs do. So what are then common alternatives, and what are things we actually use in the routine diagnostic lab? So this is minimal inhibitory concentration determination with microdilution assays. This can be even automated to a certain degree, like Vitek2, but we have to be careful in Vitek2. This is an inferred MIC, so it's not an actually determined MIC, because you only have 3 measure points and then you extrapolate the MIC, so this is really vague. Then the E-test measurement, which most of you may know. And then obviously inhibition zone measurements with disk diffusion, which you can also see here on the screen. The challenge is that these methods, they produce variable results between the methods. And you can see this here in the lower graph. On the x-axis, you have the individual measurement of the method, and on the y-axis you see the standard broth microdilution assay. And when you look here into the distribution of the individual methods, you can see, for example, on the very right, Vitek2 has the lowest concordance with this particular method. And here you even have a very major error rate of roughly 30%. So in 30% of the cases, you would say this is susceptible, but in reality, it's resistant. So if you now compare whole genome sequencing data against Vitek2, this is a classical garbage in, garbage out problem. So you really need to be sure which drug, which bug do you actually look into. And it's much more work than just pooling together more or less blindly all the data and say, here is a phenotypic method. This is completely comparable with each other, and then use this. The second challenge is the breakpoint interpretation with SIR. The SIR interpretation is very helpful for clinicians, but there are 2 different flavors of that. It's UCAST and CLSI. They also don't agree with each other. So if you take, for example, Piptazo, you have different clinical breakpoints. And this is, for example, CLSI. In the board of CLSI, there are pharma companies, and obviously this kind of influences then the clinical breakpoint discussion to a certain degree. Then we have changes in the clinical breakpoints. UCAST is every year updated. And so it can be that the SIR category changes from one year to the other. And you actually see this in national surveillance programs. When you look at the SIR category, all of a sudden you see a jump of, let's say, 10% more resistance. And that's an effect of an update. It's not that you have just from one year to the next a 10% increase of your antibiotic resistance. This usually in a country only goes up 1 or 2% per year. So these are clear challenges. And then maybe the last one is that the clinical breakpoints, they estimate a response of a certain antibiotic drug dose in the target body site. And there also is the question, can we infer from a gene, can we actually translate that into a PK/PD model? So when we benchmark, I think it's very important that if we look into these categorical interpretations like the SIR categories, what is the categorical agreement? Then the major error, very major error. There are even FDA rules, actually, how close the method has to be, what is the variability you can accept there. And then if we focus more on minimal inhibitory concentrations, what is most commonly used in the literature is plus minus 1 to 2 step difference. And this is also what you commonly see in the literature when you, later on, we will see an AI paper addressing that. At the end, what we need are very good databases where we have the phenotype, with high quality, measurement data where we are sure this is a good source and not like a research lab, and no offense here, but maybe a PhD student measuring the MIC, not following an ISO standard. I think that's very, very important. And there is a lack of these curated, and that's the other problem, the unbiased data sets. Most of the databases we have access to are just focusing on the fancy stuff. So there's a lot of resistance, obviously resistant genomes around, and not like the boring things are not sequenced. So for example, pan-sensitive E. coli, who is sequencing this? Not too many people, and I think that's a very important other challenge. So many colleagues assume that all the phenotypes are the same, but this is not the case. Then there's a considerable heterogeneity in the molecular mechanisms causing AMR, and I think that's really well known to everybody. The molecular mechanisms can be target site modifications, porin loss, efflux pump activity, enzymatic cleavage. And most tools cannot address all of these aspects. And the other thing is that the molecular, just the presence of a genetic mutation does not, for example, tell you if the efflux pump is active or not. Just because you have the gene, you don't know how active it is. So that always brings in a certain level of uncertainty. And then there can be obviously different reasons for, or different types of resistances, if they are intrinsic, acquired, or evolved resistances. So it's very hard to capture this biological diversity. Now, what tools do we actually have available to address this? I mean, the aim is to infer resistance in contrast to a phenotypic test for sensitivity. So sensitivity to detect a certain resistance mechanism is very important. And there are tons of tools out there, as we know, and they were recently summarized here in a nice paper. And the main tools which are used are ResFinder, AMR Finder Plus, and CARD. Now, the problem with ResFinder, at least when I checked the website, it was last updated in March 2024. So it's already more than a year not really updated. So, and that's one of the problems, is that some of the tools, they, I'm not sure why, but I guess loss of funding or not a sustainable case behind it, and then they start to slowly die. So I think that's a very important point. And then if you identify, let's say, an AMR gene or a point mutation, the question is, how do you translate that then into, or interpret that into a category, or let's say an MIC? And this is why the AMR rules are here, and they will obviously evolve in a certain direction and help us to interpret these findings. There are tons of papers addressing and validating different tools. I will not go here too much into the detail. I think what is very important when I checked here all the papers, or not all the papers, over a handful of papers. First of all, most of the papers are focusing on individual strains, so there's not like an overarching validate. It's a few exceptions maybe addressing multiple species, and the numbers are sometimes quite small. You see here for example, for Neisseria gonorrhea, these were 290 isolates, and then the question is, how good is this? Then obviously you have the cryptic consortium with over 12,000 isolates. That's obviously a completely different game here. So the scaling of these databases is another aspect. I will keep this short. Then we talked a lot about, validation and accreditation, and I think the purpose of accreditation or how you actually do that would be that you have standard operating procedures, quality management in place, external quality assessment, ring trials. But for these type of questions, there are not like ring trials around for genome phenotype predictions, or at least not yet. And this is the reason why there are not so many ISO accredited tools, and there are a few. I list here two. Abrit AMR and Immense are two examples from Melbourne here, obviously this one. And I think what they did is they did something extremely smart. I really have to say that. I realized that when I was reading the paper. The first step they actually did is they compared it to the performance of a PCR. So they took very clear genes, they had genes, and then measured, can we also find these genes? So it's not a phenotypic correlation in this sense, but they looked first for a certain gene. And there you can see that the sensitivities and specificity, and I highlighted this in this second line here in the green box, are very high. It's 99.6%, for example. So that's absolutely fantastic. And then when they inferred that to an AST phenotype, then you can see it slightly dropped, but still it was very, very good. And in this validation essay, if I remember now correctly, there were over 1,000 isolates were tested, so a quite substantial number. And you can see here the performance, for example, for the genes on the left side. On the figure A, subfigure A, you see MEC-A as a comparison, so MRSA detection, so this was perfect. Then VAN-A and VAN-B for vancomycin resistance was also very good. Then a few exceptions with some of the gram-negative bacteria, some ESBL and carbapenemase genes, where there was an issue, but really minimal numbers, and that explains the 99% sensitivity and not the 100%. And then on the right side, you see the phenotypic. There, the error margin was slightly higher, but I really think they did a great job. The problem of such a database, and that's the same also for the Swiss database, is that I would assume that these genomes they included or isolates they included are here from this region of the world, so Australia and maybe some of the Asian Pacific part, but it's questionable if this tool also shows the same performance with European strains. And I think this is very important, and I would really advocate this here at this conference, that we start to share strains to validate such platforms a bit broader, and I'm very happy to ship around isolates if you need some from Switzerland for validation. Immense, that's another platform. I will show you how we validated that. I think we mentioned that a few times now, what this is. The validation here, we had basically different pathogens. This is the VRE data. And you can see here on this colorful graph, on the very left side, you have the MIC, so that's the second row. And you see here this dashed red line, that's the clinical breakpoint. And as soon as you have a VAN A, VAN B gene, then it's clear that this is above the breakpoint. So there I think it's a very clear determination if you're resistant or sensitive. We had very few issues. And we compared here 14 different tools initially. And the one which was worst is actually the AI tool. So that was quite interesting. Now you can go into more complex aspects. For example, if you look for not gene presence and absence, but single point mutations. And this is an example called porin predict. Here we looked into the OPRD pore, and you may know that this is the porin Pseudomonas aeruginosa, which is the main entry for carbapenems. And you see on the right a phylogenetic tree of about 2,000 isolates which were around. We found 15 different variations, and what is really remarkable is that even if you don't have an OPRD mutation and you don't have a carbapenemase, you see here on this side that there are still some resistant isolates, despite the fact that we don't have an explanation for this. And the other way around, if you have an OPRD mutation, you also find sensitive isolates. So this genotype-phenotype prediction is not always working. Now, one thing can be the presence of an efflux pump, but even if the efflux pump is there, we still had sometimes susceptible ones. Now, does AI help us? The problem is if you have data which is not obvious, you can do as much AI as you want. You just give it a different color paint, and then may look like a unicorn. And there are a few very good papers with AI usage where they go for MIC prediction. This is one paper I found. There they looked into strep pneumonia and trimethoprim resistance. They compared a series of models. They first did a bacterial genome-wide association study. They found 2 point mutations in 2 important genes, and then could, with an AI model, predict the minimal inhibitory concentration with an accuracy of 86%, plus minus a 2-fold dilution factor. So that's very, very good, I think. It's not there yet completely, but it's very good. So, prediction of AMR from genomes, are we there yet? I personally think not yet. For some selected cases, yes. But in the clinical world, I would be careful with making these inferences. The thing is, the key message you should take home, don't underestimate the importance of the phenotype. I think that's very, very important. Where is our data coming from? Then I think likely there will not be a single tool which captures everything. I think also that's a take-home for me from this conference. I mean, there were fantastic individual specialized softwares used, and maybe you need to then bring them together. There are many, many tools around. You have to find out which one is the best for you and which works well. And then obviously to validate these tools will take a long time, because you need to do this basically for every drug and for every bug in a combination, like what was done with Mycobacterium tuberculosis. So a long way to go. But I think we are there, or we are moving in this direction. And with this, I would like to thank you all for your attention, and I'm happy to take questions if there's still some time.
Thanks, Adrian. I agree, we're not there yet, but I'm hoping that we'll get there eventually. In AMR rules, we've also sort of, noticed this issue of, there's sort of 3 main databases with sort of varying levels of curation. Do you think it's time we all sort of get around one database in some way, sort of bring the best bits of the 3 together and find some way to move forward?
I'm not, well, it could be a way to do. I think the important thing is, what is the baseline of the database? So if the phenotype method, if you would select, for example, for data coming from reference labs, or I think ideally the UCAST people, where they have all the ECOF data and all the distributions of thousands of isolates, these isolates should be sequenced through, and then you have the perfect distribution against the reference method. And that is very expensive to do, but I think that would be a fantastic effort. It was maybe a bit too European, my perspective. So CLSI probably has the same. So I also, yeah.
Okay, thanks for a fantastic presentation, as usual. I'm just thinking, do you think we could come closer to a yes if we instead aiming for RNA sequencing from the isolates or preferably directly from the patient sample?
I would say, so RNA sequencing from an agar plate, I would rather say no, because that's a completely different environment to the natural situation where the bacteria is, and I'm pretty sure the gene regulation would be very different. And to do this from a patient, maybe yes, but you would need to build like very new databases. And I mean, metagenomic sequencing or metatranscriptomics in this case could be an interesting option, but there you might also run into sensitivity issues depending on where you look. So for example, if you take a sepsis case from the blood or so, the bacteria might be too little to actually do this straight away, yeah. Thank you very much.
So thank you for all the wonderful presentations for session 4. We now break for morning tea. It's 11:20, and we'll be back for session 5 at 11:50. Okay, see you guys. Enjoy your morning tea.
Welcome back to our final scientific session of GMI15. We still have lunch and the closing ceremonies afterwards, but this will be our final set of talks. I just want to stress how wonderful a conference this has been. I have thoroughly enjoyed myself, and I just really deeply want to thank the organizing committee. They've done a wonderful job. They've set the bar so high for future GMIs. Thank you to everyone on the committee, but special thanks to our hosts, Tuyet and Torsten. Thank you so much. Wonderful. So it is my pleasure to chair session 5, which will be about surveillance ecosystems and policy environments. We have 5 wonderful speakers for the rest of this morning and this afternoon. As we found out yesterday, our Africa CDC colleague was unable to attend, so Francis won't be here to present, but that's okay. We might have some extra time for questions. So I think everybody knows the drill now. Speakers get 10 to 12 minutes for their talks, a few minutes for questions, and the bell will ding when your last few minutes are approaching. All right, so to kick things off, or actually before we start introducing speakers, a few quick messages about the session. So post-pandemic, there has been a rethinking of approaches to surveillance to address fragmented and insufficient capacities. This session will focus on surveillance ecosystems and policy environments, and takes a step back to look at a bigger picture on what policy, governance, and economic considerations are needed to establish and sustain capacity. Our first speaker is Dr. Sonia Sia, and she leads the team which implements the national routine AMR surveillance in the Philippines. Her team has successfully integrated genomics into routine AMR surveillance, and they are implementing genomics in several AMR projects. Dr. Sonia will be talking to us about sustainability of country-level capacity. So Dr. Sonia, over to you. Thank you very much. And no, just the green button.
Just this one? Yeah, that one. So it's me again. So good day to everyone. Just a gentle warning. You may or may not hear repetitions of some principles and insights that earlier speakers may have mentioned already. For this, we ask your patience. Also, a disclaimer that we are not necessarily experts on sustainability, but what we can share would be our journey to ensure that we can continue on with genomics beyond our collaborative projects, which has helped us build the competency for pathogen genomics. So, as described by Dr. Tim Dahlman, previously described, the goal of the WHO Global Genomic Surveillance Strategy Guidance document is that genomic surveillance is strengthened and scaled for quality, timely, and appropriate public health actions within local to global surveillance systems, with countries at the center of the strategy, ensuring investments are targeted where they are most needed, and with attention towards sustainability. With this as context, allow us to share our journey of integrating genomics into the national AMR routine surveillance. The Philippine ARSP, or the Antimicrobial Resistance Surveillance Program, is a lab-based AMR surveillance with 28 Sentinel sites and had been implemented for more than 30 years now, generating AMR data for the country based on phenotypic AMR data. The capacity development for whole genome sequencing in our laboratory was made possible by two collaborative projects. The first one being the Sequence project, which was a Newton-funded project with the Wellcome Sanger Institute from 2015 to 2019. And this was followed by another project, the Global Health Research Unit Genomic Surveillance of AMR, with the UK NIHR, or the National Institute for Health Research. And competency for whole genome sequencing for our surveillance, both wet and dry lab, was developed through a series of technology transfers or trainings, and then followed by provisions of requisite infrastructure, and finally, the conduct of large-scale sequencing of retrospective isolates. And leveraging on a defined surveillance system architecture that includes the usual components of a surveillance, like data collection, data transfer, processing, analysis, storage, security, and data dissemination, and data architecture that follows the system architecture. Busy slide, don't worry, we will not go into details, but having defined the surveillance data architecture allowed us to clearly visualize how to integrate genomic sequencing into the surveillance system. And guided by defined surveillance objectives, genomics was incorporated first as part of data processing as an added testing, bioinformatics as part of additional data analysis, and the genomic data to be incorporated into the surveillance annual report. So outside of the logistical support from the collaborative projects, for which we are very much appreciative, as an initial integration of genomics into the surveillance, we first focus on genomic characterization of isolates with emerging resistance phenotype, with an anticipation of sourcing through our government funds an estimate of less than 50 isolates per year. This is an initial efforts. And of course, we got less than 50 isolates, so we were able to tide over our year one efforts. As a succeeding effort in integration, a protocol for a SAT-scan and whole genome sequencing enhanced outbreak investigation was developed and implemented. For this one, we also had a secondary objective of involving our surveillance participants into genomics. In this protocol, we run WhoNet SAT-scan on data of surveillance sites to identify any clustering of isolates, get the cluster isolates to be sent to our ATM, and sequence these to confirm whether the clustering is potentially an outbreak or not. And our most recent effort towards integration is to sequence a representative number of isolates from our surveillance sites in order to have an ongoing characterization of resistant as well as some susceptible isolates to allow for tracking of emergence and spread of resistant isolates in the country. So notwithstanding the limitations in our setting, we feel that factors which facilitated integration of genomics into the AMR routine surveillance include the following: an established surveillance network, a moderate surveillance and laboratory capacity, collaborative projects which enabled capacity development for our team, and optimizing whatever available resources to meet certain surveillance objectives. And concomitant with integration of genomics into the surveillance, to ensure that we get requisite support for its sustainability, we leverage on opportunities provided by the passing of a National Action Plan on AMR and the country's engagement in the joint external evaluation on the international health regulation, where member states are encouraged to develop capacity to conduct advanced molecular testing. So from a laboratory standpoint, and just as a way to organize actions that we have taken to ensure sustainability of genomics into our surveillance, we group the factors or the actions we have taken as pre-analytical, meaning the prerequisites to the process of genomics; analytical, referring to actions to sustain genomic sequencing; and post-analytical, meaning the actions we have taken referring to matters after genomic sequencing. So we needed to be clear on the surveillance objective that is to be enhanced by genomics. We also had to ensure that we maintain a strong surveillance network, and we have to engage the surveillance participants in conversations on pathogen genomics. Thus, we conducted several digital epidemiology workshops for them, and in our annual meetings, we would include discussions on genomics. For the wet and dry lab capacity, we sent for training staff complement with permanent employment in the reference laboratory in lieu of project staff who may be with us only for the duration of collaborative projects. This is a way for us to retain a competency, a staff competency. So beyond the life of collaborative projects, we pitch for the inclusion in our annual budget for the logistical requirements for sequencing, and we are happy to have received approval for our budget request. Thus, for fiscal year 2025, our approved budget included sequencing for a certain number of isolates. However, for 2026, there was a general budget cut for our ITM, thus we anticipate to receive a relatively lower budget and will be sequencing only fewer isolates. But the management has assured us that they can look into how budget for additional isolates can be provided later on in the year. As investment in genomic surveillance is substantial, we needed to ensure that we have outputs to showcase. So we included genomic data in the surveillance annual reports and plan also to submit manuscripts for publication. So we just show in the photo our annual report and some pages where genomic data is incorporated in the annual report. Further, we are aware of the need to ensure engagement of our genomic data consumers through guided conversations on genomics. So we have set up several trainings, engaging several of our stakeholders. And for example, we have also developed a pitch talk on genomic surveillance intended for high-level stakeholders such as the executive committee members of our Ministry of Health. We continue to push for this even as we had received an initial rejection of our request. In our years of surveillance implementation, we have learned, and I think many of us can resonate with this, that patience and persistence is the name of the game. So we continue on. And as ways forward towards sustainability, we continue to be open to global and local collaborations for developing genomic capacity and potential logistical and technical support. We are mentioning this. It may or may not hint on being open to some collaborations here. We will also work towards an institute-level pathogen genomic surveillance strategy in order to have requisite policy and governance support in place. So this is not yet in place in the Philippines, and we are working towards having this one developed. And finally, resolve not to lose our enthusiasm and to keep dreaming. I think I got this quote from Professor Adrian. So, we acknowledge our collaborators who have given us the requisite leg up, and also for their trust and for their patience with us through the process of competency development. And acknowledgement also goes to the reference laboratory staff, who I think may be battle-scarred and most likely battle-weary at this point. So finally, we always give due recognition to our valued surveillance site participants because they are very crucial to our ongoing work. So with that, thank you for your attention. Maraming salamat po.
Thank you so much, Dr. Sonia. I am certainly inspired and plan to keep my enthusiasm and to keep persisting. Don't go anywhere just yet. We do have a few minutes for questions. Are there any questions from the crowd? Maybe I can selfishly start. Oh, Peter, yeah.
So firstly, thank you very much. Via David and Nansen, your work has actually inspired some of our research in Africa. So thank you very much for your example. But the other thing is that in terms of in terms of like sustainability going forward, I mean, budgets around the world are under threat at the moment. And I've seen some inspiring work from US FDA on costing the benefits of genomic surveillance. Have you had an opportunity to try and replicate that and see how much, because you said for your outbreak investigation, how much money you are saving your healthcare system by doing genomic surveillance? Because that would be amazing.
Thank you for your question. And I think suggestion, because we have not had opportunity to do that, but we've heard of the tool, and yeah, maybe we can look into it.
Any other questions for Dr. Sonia? I have a quick question for you. I was very impressed by your talk, and I'm just wondering about how you are convincing decision makers to keep genomic surveillance as part of routine operations. Could you tell us a little bit about what you think is the most influential on that decision making? Is it pressures from inside the country, that help to highlight the needs, success stories? Is it the conversations that you're having? You're talking about your workshops and stakeholder engagement, or is it outside international recommendations? If you could tell us a little bit about that.
I would love to say our conversations with them, but in reality, it's the external factors. So that's why our team would always capitalize on involvement in joint external evaluation on IHR. We get invited to sit on those, and we always capitalize on those to convince the ones that approve the budget.
Right. Yeah. Great. Wonderful. Okay, if there are no other questions for Dr. Sonia, we'll let her off the hot seat. Thank you so much. All right, next up we have Dr. Patiyan Andersson. Pat is a genomic epidemiologist and senior research fellow at the Doherty Institute. He is the lead postdoc in the Oz pathogen program, and he will be telling us about establishing national governance for facilitating data sharing across public health, clinical, and academic settings. Over to you, Pat. And green button.
Cheers. Thank you very much for this, and I think I'll start here. And this room, everybody agrees that we should share data. They see the benefits of it, but as we've been talking about for 3 days now, there is always challenges that try to curb that enthusiasm that we all feel for this. Australia is no different. We have 6 states and 2 territories that essentially operate like individual countries when it comes to delivery of public health. They are funded by the federal government, but they are responsible for the delivery themselves for their jurisdiction. Also, we are spanning quite a large area. So we have the tropical north, we down to the temperate south. So there is a lot of different disease patterns, and with that comes different public health priorities that they have. Also, there was quite a big discrepancy, at least in the past, between the resources there were for genomics, with a lot of the capacity concentrated on the eastern seaboard. Also, as with a lot of other countries, a lot of the genomic progress was driven by research. And when you have a competitive academic environment, that leads to siloing of some of these activities. And this created some inertia. And we all knew this. This gave us that undeniable urgency of we need to do something. And this actually prompted us to launch the Ostracker system that Claire Solgett presented on the first day. It had been in development for a number of years, but it really prompted that we should use this. We need something that we can share, something that is secure that we can share within Australia. So this was, the work for this was largely driven by the Communicable Diseases Genomics Network of Australia, which is a network of the public health laboratories and other public health experts. So they have worked together with the government to inform the National Microbial Genomics Framework, but also around developing this system. They sit as an expert reference panel under the Public Health Laboratory Network, which is part of the national committee structure in Australia, in the Australian government. So Ostracker has supported the surveillance for SARS-CoV-2, but also has been really instrumental in resolving some of the multi-jurisdictional outbreaks that we've had for a lot of other pathogens. It is now funded by the interim Australian CDC to provide a rapid, safe and secure place for data sharing, but also allow that equitable access to computational capacity and ensuring that for national analysis, we have some consistency. We're now moving into expanding the number of pathogens, so we have national surveillance for, and these are all underpinned by surveillance governance frameworks that are developed by the Communicable Disease Genomics Network, but consulted with and endorsed by the full committee structure going upwards. So Claire presented this vision of Ostracker on the first day. So this is just a schematic where the public health laboratories are the generators of the data, and then together with public health agencies provide data into the system. There is a dedicated public health server that analysis is done, and that is carried out by the national analysis team and then reported out. So I'd like to talk a little bit about the research that underpins a lot of our decision making and what we want to do for genomic surveillance. And one of these large ones, there had been a lot of research done in the individual jurisdictions, but a large translation national project was done by the OSPATHIAN program. So this is a $10 million program that ran from 2021. We're in the last year of it now for this phase. And it seeks to really generate that evidence for what is useful to do genomic surveillance for, how should we do it, how do we implement it, and what is the public health impact. And part of that is a lot of the public health utility, the cost effectiveness of this, in order to be able to see where is our money best placed. We have a range of focus areas, and we have some model organisms that we've been working with. The structure is that right in the middle of the day, you have the engine of the program, which is the public health laboratories and their affiliated public health agencies. But also, when we shifted from just doing this for a public health purpose, but also into research that needs to be published, we need to have an ethics framework, and we needed to have a consistent ethics framework that was across the whole country. In Australia, there's now something called the National Mutual Acceptance Scheme, which allows approval in one jurisdiction to be recognized by the other jurisdictions. Of course, you still need to have governance at each of the collaborating sites, each of the public health laboratories, and the public health agencies needed to have their approvals as well. But it fast-tracked the system and allowed us to have a unified framework. The second part is how we have worked. We have pathogen-specific working groups that is a group of people that are multidisciplinary, so it pulls representatives from the public health labs, from the public health agencies, academic expertise, to collaborate and work together to define and execute these projects that we have put up for these pathogens. And that includes defining what sequence, what metadata should go in. There is a terms of reference that defines how the data should be used, so people know what happens with the data when they contribute it, and also that includes authorship, which is always a discussion when you are looking at academic outputs. And then we have the third part, which was OSTracker. By this time in 2021, we had used it for a year, and it was a known entity to a lot of the people who work with this. There is a considerable overlap between the people who are supporting us in the collaborating in this research part and the public health delivery. So it was something that was familiar to all of the partners. It needed to be adapted for a research perspective. We needed to be able to ingest sequences from a wider range of organizations, so that we needed to work on the sophistication of the permission system for these partners that were part of it. This was the original OSTracker that we used for public health. We need to keep the data separate. So the research data and the public health data should not be overlapping in this case. So we compartmentalized the data, but also compartmentalized the analysis. So we have a specific analysis server that people could collaborate on that is different from the one that we use for the public health, so that we can have this collaborative environment to work in. There is formal data contribution and access agreements that are signed by everybody that touches the data or contributes the data, so everybody knows exactly what happens with it. So this was a trusted system, and for more than 200 collaborators across 45 organizations. And a part of this was that we needed to enable everybody to contribute. So there was a lot of capacity building, both in terms of hardware, but also in terms of positions, so that each jurisdiction could contribute the data and be part of this. So the success was that we have collated 45,000 sequences, and this does not include the SARS-CoV-2 data, so this is just for all of the other pathogens. And also we have had huge developments in the relationships across sectors for the One Health area. So what we have now is that we have created an environment where you have translational research for pathogen genomics using the same platform as the public health service delivery. So it means that any findings that we generate on the research side are very easy to implement and apply into public health. And a strong part of this is that we have been working very closely with the federal policymakers. They have been involved in each of these areas. They are part of the executive level and the working groups for the research and provide advice into there. They fund the OSTracker system, and of course, they are the main users of the public health data that is generated on the other side. So following a stepwise development since 2015, which was when CDGN started, we're now at a significant stage in Australia for public health, with the establishment of the Australian CDC. And they will need continuous evidence to base their policy decisions going forward. And we have now received continued funding for another 4 years for Patiyan for the second phase. And if you want to have more details on the establishment of the first phase of the program, you can use the QR code there. But through this program, we have developed a strong network of collaboration across all of the different jurisdictions and across sectors. And we believe that this will continue to strengthen as we go forward. So I'll come back to this, but that we should share it both nationally and internationally. So the data sets, which total 45,000 genomes, will be a highly valuable resource to both contextualize future public health investigations, but also underpin future research. And all of the partners, as Jake mentioned yesterday, have agreed to that all of these genomes that we have collated should be made available for the international community and uploaded to NCBI. So I have the privilege of standing here talking on behalf of a lot of people, both from Australia and Patiyan. So I'd like to just reach out to thank you to you all from all of them.
Thank you so much, Pat. That was really, really impressive. There are so many different working parts, going on in the program that you described. Are there any questions for Pat? Yes, Adriano.
And sorry if look, my question means that I don't understand something. But is in this framework also the Organization for Food Safety included, and or not, or just public health organizations, institutions?
So in Australia, we have something called the AusFoodNet, which is our network that sits under the public health national committee structure as an expert advisory panel for the community with diseases branch. They deal with the foodborne investigations, and they are linked with our food safety authority as well.
So you are able to do clustering and matching between clinical cases and food samples?
So far, in some of the outbreak investigations, we don't have a formalized ongoing surveillance for that. But in some of the investigations where it has been appropriate for Salmonella and sometimes Vibrio, Parahemolyticus, there has been some samples that have been agreed upon that they could be included from non-human sources.
Thank you.
I'll just follow up a little bit on that. I mean, a lot of our laboratories in Australia do both, so they test in food as well as human samples. So there is integration of food and human data in the platform. Yes, so it's not the same in every state and territory. And then the other regulator that's included is the Food Standards Australia and New Zealand, which has sort of been invited to be part of the working group. So they sort of have input into their policies around genomic surveillance.
Other questions? Yes, over here.
Thanks, Pat, for a nice presentation. Have you been able to build in some kind of outbreak alert system? And are you allowed to share data between the states in real time?
Not yet, because we don't have for many pathogens, we don't have ongoing surveillance, but that is something that we are working towards, and we are building the frameworks for that. And in there would be the vision of having, if it's appropriate for that pathogen, some sort of alert where people can upload, there's a regular analysis done, and then people who contribute data will be alerted to that there is a match or something like that, and there is a recommendation for further follow-up.
In terms of the surveillance, do you have any sort of sampling strategies? Or whether do you work with the other part of Australia in terms of the exchanging the samples or the isolates among the laboratories?
Yeah, thank you. That is the large part of Patiyan, is that we want to get a national view of what the situation looks like, so that we can have the best available data to be able to design our sampling strategies for what would be appropriate for a national surveillance.
Yeah, one more thing, it's really, how do you really collaborate or share your data with the other countries? Do you have any sort of a policy or strategy in terms of the sharing your data sets, or how do you really incorporate with other countries?
Right now, as from the research project, there has been agreement that we can share this data into international projects, so there's no formal exchange of data. We're hoping that this has, as Jake was mentioning yesterday, that we're hoping that people have started to think about that sharing of data is beneficial, and there is the benefits outweigh the risks of it. So we're hoping that through this process that we have started to change that mindset of.
I think there's one more question over here.
Thank you. Wonderful presentation. Do you have cholera genomic data in your collection? If not, do you have any plan? And I have one more question. That is, did you sign memorandum of understanding with the collaborating institute that contributed all these genomic sequences?
Yes, so there are collaborating agreements between each of the institutes that contribute data, and they contribute both sequences and metadata to this. So there's a very formal agreement through this research collaboration. On the public health side, there will be, everybody has to agree to the frameworks that we are proposing and endorse those. In terms of the cholera, a lot of the public health labs will have been gathering these such samples and analysis of that. Right now, it has not been analyzed in Patiyan, but it's something that, should there be an increased need, that we can continue.
So you have any cholera surges in Australia? Do you have any?
I think it would be mostly return travelers that would we have. So there is no endemic transmission, so it would be just people who have been usually exposed overseas and then returning to Australia and diagnosed there.
Thank you.
Oh, Mark, you have a question. Oh, okay. Wait, hold on, Pat. I have one more selfish quick question. So there are a lot of different partners involved here, and that's a lot of consensus building. So I'm wondering if you have any tips for us about relationship building, or whether it's just like pure mojo.
I think getting everybody in the same room and allowing them to be able to talk. This is something that Ben has been leading for a long time through CDGen. So the public health laboratories have been working for a long time. They have brought in their respective connections in the public health world. So I think it's just grown from a very strong core of people who wanted to work on this, and then it has grown and tried to spread the enthusiasm.
Fantastic. Yeah. Thank you. All right, wonderful. All right, our next speaker is Marc Allard, FDA. He is a distinguished research microbiologist at the U.S. Food and Drug Administration Human Foods Program. He has a PhD in biology, and Marc has established himself as a leading expert in the fields of genomics, bioinformatics, and molecular biology as applied to food safety. And Marc's going to tell us about foodborne pathogen surveillance and economic return on investment estimates for genome tracker laboratories. So over to you, Marc.
Thank you. Thank you very much. Without further ado, we'll get into this. So we've been asked to evaluate costs and benefits of WGS, and there has been some literature. In fact, Masami was one of the first people that said, we really need more tools for FAO and all their member states, and so who's going to help? And but there have been some early papers, Altwelt et al. In 2021, and I'm pretty sure there's several of the original co-authors in the room here. This was a study of cost-benefit, and essentially the key information, the costs, are the costs for the equipment, the supplies, the staff, time to do both the wet lab and dry lab analysis. And then the benefits are really the economic value of avoiding illness. This is the kind of data that we can get our hands on. And so what we did is essentially replicated and updated Altwelt's model for U.S. public health labs. And this is just developing tools to assist the labs to put their cost data into like a spreadsheet, and then we'll have a piece of software that takes that data and converts it into return on investment kinds of information. But there's nothing really new. I mean, we're standing on the shoulders of the giants that led us forward in these topics. But the plan is to develop a tool to do the quantification. This will actually be, we're hoping it'll be a shiny tool, but we're still fussing with computer fun. Maybe Torsten has some suggestions for us. So the data source, the data is just Genome Tracker. It's just 3 pathogens, Salmonella, Listeria, and the Estech E. coli, and it's only samples from food, animal, and the environment. So some of our earlier work, we took like all of the CDC data and all of the USDA data, but this is, we're trying to just say, this is a time of resources, and so we got to sing for our supper and show the value of what we're doing. So the cost breakdown is broken into equipment, consumables, staff, and we threw in an other just to give them options. And these are tabulated on a per sample basis. So we're trying to convert those numbers to the actual cost of isolates, which comes right out of Altweld. And so the benefits are the cost per illness plus illness avoided. And this is essentially, and this is in US dollars. And so there's a study, Hoffman et al., which calculated the cost of an illness within the domestic US. And what you see is there's different impacts for different pathogens. Essentially, the more virulent and deadly you are, like the Listeria, those actually have $2.5 million per case of avoided illness, and it's because of miscarriages and deaths, primarily. The second piece, for estimating illness avoidance comes from Brown et al. And so this is a study we published in 2021. It was a model to look for the correlation between the numbers of sequences and the numbers of illnesses and outbreaks. And what that study did was it shows that for every 1,000 isolates sequenced, you're reducing 9.09 or 6.09 cases of illness. And so those can be tied to the calculation. But this is where these are the information you need if you want to do these kind of studies. You'd have to collect some of this yourself, or you make the assumptions that the way things are working in the US are similar to other places, but we know there's different food culture and differences around the world. So to get into it specifically, the equipment costs are the gear, the sequencers, any major equipment, and we also are annualizing this capital cost across the life of the instrument. And we're also asking that, and there's also often a maintenance cost that's part of that. And then we added the user rate, because many of the labs don't just do their sequencers for the food and the environment, they're doing it for other species, they're doing it for clinical, they may be using their instruments for other surveillance tools. Consumables are pretty straightforward. This is all the consumable, Petra plates, pipettes, and things like that. And then the staff costs are essentially, this is what we're calling is the actual time, hands-on time, and that the staff are working not only to collect the data, but to report on it or to analyze it. And so what we did was we took average pay rates that are also within US, they can give different classifiers. So this includes both a technical cost, staff cost, as well as like a senior scientist person that's overseeing and actively working with maybe involved in interpretation. These are, some of these, it's always tricky. Do you want every region to tell you all their costs? Some may not want to share that. They may all want to move to Melbourne or leave Melbourne because of the cost and the pay. So by using a standard cost, you can integrate it. And of our 31 labs that are currently in Genome Tracker, 14 chose to work with us and collect data. And so here's the basic rundown. I'm giving you the averages. So the average equipment cost is $156 per sample. But there's a range that the labs show $14 to $395 per sample. And so there's quite a large range. Consumables is $175 US, staff time is $47, and other costs are $13 for a total per sample cost for the average of just under $400. And the fact is, if you go and read Altwell et al., these numbers aren't that different. You know, if you add in cost of inflation, and you see this broad spread across the different laboratories. Now, we also looked at the numbers of sequenced annually per lab, and that also varied a lot, from $53 to $400. 53 to 400 isolates sequenced per year, with an average of just under 200 isolates. So these are mostly low throughput, and it's partly because getting a hold of environmental and food isolates is a lot of work to collect that. So these are modest, and like I say, there's significant variability across all of these based on labs. And I think Altwell originally suggested that it was economies of scale. Essentially, the labs that sequenced more got lower per sample costs. And also, if you have bigger capacity instruments, sometimes that reduces the cost. There are also ways you can like dilute your reagents and do half reagents or other strategies. So here's the preliminary results for cost benefit. And what we're doing is we're just pushing those numbers through the system. And I showed you the papers and the data that it's based on. And so the number of salmonella avoided cases was almost 300 cases. And if you calculate that by the per case cost, it's 4.937 million in averted illness just for salmonella alone. Oh, I don't know if I mentioned, we also do a multiplier piece. The CDC says for every person that reports sick for salmonella, 39 people don't come to the hospital. They're just at home. And they wanted to capture that. And so we used those amplifiers in accounting of the cases. For Listeria, there are only 5 cases avoided over the last year, but that's over $11 million of averted illness. And then the Estech E. coli's, it's a more modest 733,000 for a total of $17.156 million for about $1 million of investment to sequence about 3,000 isolates. So a little bit less than 3,000 isolates. So when you do the cost benefit analysis, what it says is that for every dollar invested, we're saving $17 in healthcare costs. And actually our study, Brown et al., we said 1 to 10. And so we've actually improved over the last 5 years. But I honestly, I can't tell you, did we really get, if it's, the last time we calculated it in 2019, for every dollar invested, we saved $10 in health cost, and now it's 17. So, but I don't know if this is just a high-performing group of 14, or it's maybe, taking it just the food and environment, it's more impactful than, or maybe it's less. We can't answer that piece of it. So their planned outcomes are, 1, to publish a study detailing the details of states and their throughput and their return on investment. So that once we get all the I think we have like 24 or 25 labs that said they're going to send us our data, but we've got 14 labs to present today. I wanted to present preliminary data. The second is that we want to produce a little piece of software that does the cost-benefit calculation so that labs can use these tools. They can plug in their data. I mean, I think all the labs know what the cost is for their reagents and their staff. It's like we just heard, you got to put together a budget. And so these would be known. And then we plugged in the different model values, and then we could get information. Oh, you make me worry about the button? I wanted to say another outcome. I'm already in conversations with Masami, okay? And that is, should we have a workshop to show people how to use this? You know, maybe next year we could have every GMI member who is willing to volunteer information, and we can give our first global impact study on the value of our networks. This would be a nice working group, and we're here to help with analysis. I just wanted to show you the publications. Ford et al., Australian, he said they had similar cost-benefit, where it's equal cost-benefit is that if you can reduce illness by 2%, then the cost of building the lab pays for itself. And the earlier study said it was more like under 1%. But the studies are here, and then there's references, and I'm happy to take questions.
Okay, lots and lots of hands. Okay, Eric first.
Oh my God, I've been waiting to do this for so long. Eric Stevens, Hygiena. Interesting question or presentation, Mark, from FDA and what they're doing. I was really curious when you're talking about a cost analysis, especially considering some of the challenges with data sharing, trying to also model that to the availability of data. So really getting a good argument for seeing perhaps a decrease in cost per sequencing or an increase in cost savings by the availability of data. So if you see countries, in the US you could certainly see that, so as states joined and were uploading their data, how much return on investment was that? Of course, that'll be a little bit tricky because at the time it was a little bit more to sequence back then. But certainly going forward, I think that would be a great kind of impact too, and being like, we could save money and lives by sharing data.
Right, I'd also like to add that you have to act on the data. So if you see a cluster for food safety, you have to get the contaminant out of the food supply. You have to stop transmission, and that makes more people not get contaminated. And that's also the reason we should globally share is you may know about a problem, but the sooner I know, I can take care of it in our country, and fewer of our people will get sick. And so it's like, let's all share, because then we help each other, and that will increase the return on investment because fewer people will be getting sick. But I think to build a typical lab today is probably $200,000, $300,000 just to get the bare bones. But everybody's got their budgets, and so once again, that times 10 or times 17, if the return is that high, you know, you're averting illness. The question is, who in the government cares about that? That's willing to pay money to prevent illness for their citizens. And so that's where the message, that's where some of the politics is. Typically, the people that fund the laboratory and capacity building aren't the same people that are thinking about the number of averted illness. Oh, and if I do a quick calculation in my head, for 45,000 isolates in Austracker, if it's similar to the US, that would probably convert to about 200 million, somewhere in that area. Other questions?
So I don't have a question, but I have a comment to kind of piggyback on your GMI work group idea. I've been working on reducing costs for library prep because we all have budget constraints. We've been looking at some manual preps, and I know Adrian's got a really good automated way of reducing library prep costs, but I think maybe a work group on reducing those costs and consumable costs are also really important.
Question over here. Question over here.
Thank you for a very engaging talk. I just wondered on slide 199, where you had the cost per sample, whether this factored into, this took into account the platform for sequencing.
Oh yes, yeah, it's platform specific, and so in fact I didn't list it here, but we would also ask which instrument are you running, and if you're like some labs have multiple instruments, so you would probably want to do this calculation separately for each instrument because they would have all those costs could change, consumable, hands-on time. But I also think like this could be used like, let's say a lab says they want to go to new capacity. I want to get my new I-100, okay? Well, we could calculate their cost and see how much money does it save you to buy an instrument. See, you save you buy by saving, so it helps.
So you saw difference between short read and long read, and what was the difference?
I don't have those details worked out. That's a very good question and something we should do.
Okay, we have 2 more talks to get through, so we're going to get one more question, and then I really encourage you to come and clobber Marc with more questions during lunch.
Okay.
Okay, there's a very strong hand over here.
Hey, this is Angela Potes with APHL, and just kind of piggybacking off of that comment, was wondering if you all were aware of or had utilized the genomics costing tool at all from WHO when you guys were looking at this and doing your cost
I'm aware
estimates.
of it, but we decided to use the Outwell strategy. But you're absolutely right. Exactly how you count this, and I think the more we can get, so we're making sure everyone's counting the same thing, and we're not getting diversity because of interpretation, that it would be extremely valuable. And so the tools from WHO and FAO we're happy to look at.
Yeah, and so the first version was SARS-CoV-2 based, but the second version takes into consideration and has the option for enterics, bacterial, other viral. It considers the different instruments and if you have more than one. So it might be really useful, and I'm part of the working group, so would be happy to talk about moving forward.
Thank you.
Wonderful. Thank you so much, Marc, and thanks everyone for your questions. Our next talk is by Ines Atmosukarto. Dr. Ines is a technical consultant for the Australia-Indonesia Health Security Partnership and currently acts as a technical advisor to the Indonesian Minister for Health. She has been working closely with the Indonesian Ministry of Health to support their health biotechnology transformation agenda, and as the architect behind the recently launched Ministry of Health National Genomic Initiative since 2022. So Dr. Ines, here you are. She'll be telling us about establishing sustainable pathogen genomics in Indonesia, where we are at now. Over to you. And green button here.
Thank you very much. And I'll start with thanking the organizers for the opportunity to share Indonesia's experience, but also with a disclaimer. I'm neither a bioinformatician nor a clinical microbiologist, so I'm more of a generalist and a crazy person who, at the end of the pandemic, answered the Ministers of Health challenge on what are we going to do with all of these sequences that have been donated to Indonesia by the global funds. So during the pandemic, there was this huge importation or donation of genomic sequencing instruments, which came late, by the way, which came 2 years too late, ranging from like small machines like MinIONs right all the way to T7s like from MGI and Novasix from Illumina. So obviously the challenge was on what to do with these instruments and how to use this opportunity to actually accelerate genomics in Indonesia. And it started with 10 slides sent to the ministers, and he challenged me to stay on and actually drive the implementation. So Indonesia is a huge country. It's an archipelago country, so that brings challenges. It spans 5,000 kilometers from east to west. So immediately you can see the challenges associated with doing anything in Indonesia. The logistics are quite challenging. If we look at the diseases that are issue, causing an issue in Indonesia. You have the non-communicable diseases, reflecting the diagram or the graph showed yesterday, where we have a rise in the non-communicable diseases, but we still have issues with infectious diseases and tuberculosis, although there has been significant improvement compared to 2009, still remains a significant cause of mortality in Indonesia. So that's been the first challenge in the infectious disease space. So just to put this in context, there's an estimated a million cases of tuberculosis in Indonesia. And even 3 years ago, less than 50% of these cases were actually identified. Through a huge drive in active case finding, we've seen that jump to 856,000 cases identified last year, and about 12,000 of those being drug resistant. And with this increase in detection and the identification of drug resistant TB, we saw this as an opportunity to actually implement genomics for drug-resistant testing in Indonesia. But that's in the actual framework of a much bigger program, which actually has a much bigger focus on non-communicable diseases, around 9 areas of interest that are actually under this new initiative that was launched in 2002. So over the last 3 years, Indonesia, or this initiative, went from having 0 sequences in its database in general last year to having about 12,000 human genome sequences in the database, and growing an increasing number of TB sequences. And TB is the only infectious disease at the moment covered systematically through this initiative. So aside from building systems to collect metadata and biobanking infrastructure, the initiative is also building a trusted research environment to allow access and sharing of data. So just to focus on the TB program, so this pilot program has a very ambitious target of actually doing direct from sputum sequencing on about 8,000 samples collected from the 34 provinces of Indonesia. And this is focusing largely on drug-resistant samples. So these are samples that would have been identified as being rifampicin-resistant based on the existing tests that are implemented in the country, and then being a subset of those being tested through targeted genomics directly from the sputum, and a smaller subset of that also being sequenced through whole genome sequencing. And the provinces that where we focus the sequencing from the isolates are based on the prevalence of drug resistance in those provinces. In addition to that, the program is also building whole genome sequencing information from the hosts as well, which will be available for research. So this program is actually an exercise in madness because it involves gathering samples from over 400 primary healthcare facilities. Right? So we've had to build trust with these healthcare facilities, and being a Ministry of Health-driven project has actually helped that as well. Now, all these samples, according to the national program, all drug-resistant samples get funneled through a drug sensitivity test lab. And it is that lab that then sends the samples for targeted sequences to 17 labs that have received machines from the Global Fund and which have also been trained in implementing the targeted sequencing workflows. So you can see how this is already quite a challenge, and it has taken a whole year to just develop the agreements and building the trust between these various stakeholders. Now, just to also give you a bit of a feeling for where all of these labs are located in Indonesia, and you can see that there will be some distances. So your triangles are your DST labs, and your different colored dots are the sequencing labs with either a grid iron or a MySeq machine. And these DST labs then act as the police that sends the different samples to the different places based on pre-existing agreements. Now, data analysis is done locally with standard pipeline, and to manage the data, we try to then centralize the storage of all these BAM and VCF files so that they're not lost in these various labs, because otherwise they'll probably be on a laptop that stops working. So what have we seen? So this is an analysis of what has been done on the targeted sequencing work, and this is just about half of the target of that 8,000 target, and we can see that in doing the comparison between the targeted sequencing and the DST results, the specificity of the targeting TNGS assays is quite good, and it parallels some of the published WHO references, but the sensitivity still lacks for some drugs. And at the moment, these inconsistency between the targeted sequencing results and the DST results are being investigated. And from these targeted sequencing, we've been able to actually have a big picture view of what is happening with drug-resistant TB in Indonesia. And we can see that when you divide Indonesia into 3 different regions, you can see that the drug resistance profiles of the isolates that are circulating in the western part of Indonesia are definitely different from what is circulating in the eastern part of Indonesia, and the central part of Indonesia is a bit of a mixture of everything. So even this high-level view of what is happening should actually inform the policymakers in the ministry. From that data, we've been able to generate some profiles of what is happening in the various parts of Indonesia. And obviously, this data, as someone before me has pointed out, needs to be acted on as well. So we've also identified that the 17 labs were not performing to the same level, and that there's differences in the quality of the sequencing that is done there, and we're trying to drive standardization and capacity building across these different labs. And this is in line actually with a roadmap that was published by the Indonesian Ministry of Health. Their national TB program actually has an NGS for tuberculosis roadmap, which identifies the implementation of NGS for TB surveillance and as well as for clinical use. So as this meeting has talked about data sharing a lot, and since Indonesia does have some legislations around what can leave the country and what cannot, I think one of the things that I wanted to make sure is that I said was that collaboration is actually encouraged. In fact, the whole idea behind building these databases was to encourage collaborations, but to move the collaborations away from collecting of isolates that are then sequenced in Melbourne or somewhere else, to actually collaboration on analyzing the data, because the data is there. The capacity to actually dig deep into that data still lacks in Indonesia. But by having a database or having this 8,000, data there, I think I'm hoping that this will actually drive collaborations with globally, so that we can actually build the next bioinformaticians of Indonesia. So it's not to say that data sharing is not allowed. It's actually allowed, but we're trying to minimize the data leaving the country, and we're trying to actually make sure that if it does leave the country, there's actually clear reason for why it has to do so. So the next step for this is to actually integrate this into the national TB program and to prove that this is actually useful to the national TB program. And looking at the journey of a TB patient, you can actually see that there is a lot of data being built around all of these TB patients. So there's, and there's so many systems that independently collect data. And in fact, one of the things that the ministry is trying to do is to integrate all of this data into a single platform where all of the information related to that patient is actually in that platform called Satu Sehat. So you can see that genomics is part of the data that must be integrated into this. And I think having all of this information into the one place will actually help the ministry drive better responses for each of these TB patients, because obviously this one-size-fits-all approach isn't working, and Indonesia still has a huge challenge ahead of itself, and using these tools we hope is going to help that. So this is my last slide. It's actually to say that, as you could see from our previous slides, this is the work of a huge number of people, and it would not have been possible without funding from the Global Fund and FIND. Thank you very much.
Thank you so much, Ines. That was really awesome. Questions for Ines? Is that a hand up? Okay, Tim.
Thanks for the presentation and really impressive work in Indonesia. Can you speak a little bit about the kind of development and maintenance of the bioinformatics capacity that's needed to support this?
So bioinformatics capacity is actually a luxury to find in Indonesia. It's really hard, and if they are around, places like Illumina snaps them up, and the Ministry of Health cannot compete with the salaries that are being offered. So we're trying to build capacity through doing training. So like upskilling permanent staff of these various labs, just like the Philippines, because otherwise we're just going to lose the capacity. So this sort of training is how we try to maintain them. But I think keeping them in Indonesia is hard because Singapore can offer much bigger salaries.
Lots of questions.
Hi, impressive work. Just a question. What were the challenges in terms of specimen transport logistics? Because I know for a fact it's really challenging. Like the Philippines, like 7,000 islands, and yours like 17,000. And second, do you have some findings in terms of were there discordance between phenotypic and whole genome sequencing or sequence data in terms of drug resistance?
Yes, so logistics is a huge problem. Some samples have to come on the back of mopeds, right? Some samples are transported, in other ways. Luckily, and this is really fortunate because we are under the Ministry of Health, we were able to piggyback on the current logistics for TB samples. And it was really important to not disrupt that. And this is why you could see that the samples go from the primary healthcare facility to the DST lab. That they would have gone this way anyway. And the last step going from the DST lab to the sequencing lab is where we had to provide samples. But because they were pooled in these just 12 DST labs, it allowed to pool samples and then to kind of reduce the cost of the logistics. So that's to answer the logistics, but it's always an issue and it's very expensive, right? And discordance, yes. So I think part of the the calculated sensitivity issues is like there's discordance between the results from the phenotypic testing to what we saw in the genomics. And we haven't dug down to see whether that was related to specific DST labs. I think there's a lot of more digging into the data that needs to happen because it's 12 different labs, right? And just by hearsay on the grounds, we hear that, some labs had antibiotics that were expired, or, so there's other things that may also contribute to that. So we need to really dig down into the data a little bit more.
Thanks very much. Such an amazing presentation and so much work. Just following on from the previous question, so with the TNGS, was that being done using a prescribed protocol for all the labs, or was it a commercial product?
So we used a commercial product. So we used the Dplex kit to run on the Illumina machines, and for the labs the 5 labs that were using an ONT sequencer, we used the in-house provided primers, and were very much driven by their protocol. So I'm aware that the ONT has now commercialized through bioMérieux, and that's so we did not use the Ample bioMérieux kit.
Okay, and to kind of get over those differences between labs, and actually, I guess, check the performance of the genomics, were you running a quality assurance program or an EQA?
So this is actually retrospectively being run now, because when the lab, I guess the program kind of like was building capacity as at the same time as it was generating data, and as it came out that different labs had different performances, like the number of failed runs were very different from different labs. So with the TB hub of the BGSI is now running this sort of standardization and quality control. So more will come out of that.
Yeah, that's great. And so were you able to, bring the labs together as a network to work through some of those issues?
Yes. So fortunately, with funding from the global funds, and find part of that funding actually went to bringing together all these 17 labs, 2 or 3 times a year, so that they could talk about their issues and share their experiences.
Great. Amazing work. Thank you.
Okay. Unfortunately, funding is running out, so.
We have one more very quick question here.
And I promise it's not a question. But so Terima kasih on that great presentation. When I was at FDA, I had a great pleasure of working on 2 separate projects with Indonesia, and working on kind of high commodity shrimp exports. And now that I'm at Hygiena, that's kind of a regular part of my job, where we're working with industry on understanding the root cause of the problem. And so I think really going forward, I was always impressed with the willingness of my Indonesian counterparts on what they were willing to do, to share, and my colleague Julie is really leading that effort now. And I think that's going to be a great example going forward of that public-private, industry, government, whoever wants to come kind of partnership going forward. And so at Hygiena, that kind of thing is very much like what we're into now and helping out. So it was not a question, it was just kind of a thank you for some of your colleagues. It was a true pleasure.
Thank you.
Wonderful. Thank you, thank you, thank you. Thank you, Dr. Ines. Last but absolutely certainly not least, we have James Ong. Dr. James Ong is a research officer at the University of Melbourne. His work focuses on the evaluating and implementation of outcomes of pathogen genomics in public health systems across Australia and the world. So James is now going to tell us about monitoring and evaluation of genomics implementation. So there you go.
Thank you very much, Lisa. Oh, yep. And thank you for the organizers for inviting me to present such an important topic on the monitoring and evaluation of genomics implementation. So, before I start, when I talk about evaluation, I'm not talking about evaluating pathogen genomic tools or code to see whether they work. I'm more talking about pathogen genomic programs, from collecting the samples to sending the samples off to end users to decide how the genomic data should be used. So just something to keep in mind. Before I make a start on the presentation, I'm going to actually define very quickly what monitoring and evaluation is with some specific examples. So monitoring is specifically relating to a process to periodically collect analyze, and use information to actively manage performance, maximize positive impacts, and minimize the risk of adverse impacts. And it can provide early and ongoing information to help shape implementation in advance of evaluations. So, for example, we could be monitoring how many samples we are collecting from different samples, how they're being processed through genomics, and whether we're collecting complete metadata to actually inform us of the samples that we're collecting. Evaluation, on the other hand, is the systematic assessment of the operation and or the outcomes of a program or policy compared to a set of explicit or implicit standards as a means of contributing to the improvement of the program or policy. So in other words, evaluation in a broad sense is looking at how is the program being run, is the program achieving the outcomes as expected, and how can we do things better to improve on our programs. So basically an example of this could be we're looking at how pathogen genomic data is being used by end users, and so we look at whether that's working, and we look at identifying how could we do things better to actually increase usage of pathogen genomic data by end users so that they can incorporate the data more to inform public health responses. So why should we actually evaluate public health pathogen genomic programs in the first place? Well, there are a few key reasons why we should be evaluating pathogen genomic programs, and I think as governments are as we're going under monetary constraints where governments have to decide how they should be spending their money, more and more there is a justification that we should be evaluating our programs to see, to justify the value that governments place on those programs and to ensure that we keep getting funding to run those programs. So you can see that a few different reasons for why we run evaluations include to fix up inefficiencies and redundancies, to ensure that outcomes are being equally distributed, to record unexpected outcomes as well as expected outcomes of implementation, to look at investment infrastructure and training and see how they could be improved, to measure sequencing capacity, as well as to develop guidelines and policies like what was being presented in this conference. And a framework that we use to evaluate pathogen genomics programs is the Pathogen Genomics in Public Health Surveillance Evaluation Framework, or PG-phase framework, that my supervisor, Angela Ferdinand, has developed. And broadly speaking, we can split pathogen genomic programs into three different phases. So phase one is pre-analysis and analysis, which is basically we collect samples, we process the samples, sequence these samples, and get the genomic data. Phase two is reporting and communication, so that involves the bioinformatic analyses as well as compiling the results together into a report that we send to end users. And then phase three is the interesting bit, which is public health implementation, which is where we end users, such as policymakers, public health authorities, get the pathogen genomic reports and decide how they should use the reports. So this use can range from informing clinical treatment of antimicrobial resistant bacteria to confirming that public health responses, the correct public health response has been carried forward, to actually informing surveillance and outbreak responses against infectious disease and AMR. And there are a few different quantitative and qualitative measures that we can use to measure, firstly, the use of pathogen genomics, and secondly, the outcomes of the use of pathogen genomic data. So as Patiyan has explained previously, we have the Oz pathogen program, which is basically consisting of a number of different arms. And as part of the Oz pathogen program, we are evaluating the utility and cost effectiveness of genomics-based public health responses across Australia and New Zealand. And so within the evaluation arm, we have a number of different evaluation studies, ranging from a situational assessment of public health laboratories against genomic capacity, to prioritizing which pathogens should be sequenced first, to doing analysis of case studies of pathogen genomic programs to see how pathogen genomics data is being used and what are the outcomes resulting from that, to doing an impact assessment of what is the impact of pathogen genomics on public health responses, and also doing an economic evaluation, just like Marc has presented, basically outlining what are the costs and benefits of doing pathogen genomics compared to conventional biological techniques. And we've recently published something in this area where we modeled, done an economic evaluation of a model of non-typhoidal salmonella, and we find that despite the increased cost associated with sequencing, we see the number of cases averted and reduced healthcare costs associated with averting those infections. So we are publishing some papers around the area, and you'll see hear a bit more from the evaluation studies in the coming months. So what I'd like to actually give an outline now is just something that we've been developing in collaboration with the IPSN, which is a pathogen genomics monitoring and evaluation tool. So there are a number of principles that guide the development of the monitoring and evaluation tool, ranging from aligning with key objectives, strategies, and documents to focusing on public health utilization of data and the resultant outcomes. We're also wanting to align the tool more to make it appropriate for countries at different levels of capacity, and I'll talk about the maturity matrix later. We have a whole system approach encompassing from the beginning to the end, from collecting the samples to actually sending the samples off, and sending the genomic data off to the end users, and also providing some guidance on how pathogen genomics programs could be improved, and also just reducing this respondent burden by actually spreading the information, the data out to as many people as possible. So the monitoring and evaluation tool is an evolution of the PG phase framework that I've presented earlier, which is basically an evaluation framework that splits the genomic system into 3 phases. And this tool is used in a number of different studies to look at the current status of genomic implementation and how it can be improved. Whereas with the monitoring and evaluation tool, we're looking at a capacity strengthening framework for pathogen genomics informed surveillance systems that countries can use to actually identify the strengths and the areas of improvement in their genomic programs, which can then inform ways and programs that they can implement to improve their pathogen genomic programs, and to also track the impact of those programs. So the tool uses a maturity model matrix, which is basically for each level of each part of pathogen genomics programs. They can be, and each subsection within the pathogen genomics program, a country can be rated on 1 of 4 different levels, which range from the foundation level, which is just they're starting to establish capacity in that area, all the way to development and optimization as they continue to develop the part of the program. And lastly, the mastery, which is where they've established capacity for that particular part of the program, and they're now starting to share their capacity with other countries. And unfortunately, I can't actually seem to back out. Oh, wait, hold up. There we go. So in terms of the maturity matrix model, we are undergoing development and piloting in terms of the monitoring and evaluation tool. So we've set out some priorities, we've developed the goals, we've developed the strategies, we've implemented the strategies, and we're just iterating through the different piloting of the pathogen genomics across a number of countries to actually just continually refine the tool, and we expect the tool to be available by the end of the year. So in terms of the tool is split up into 6 different parts relating to the different parts of the pathogen genomics program. So those different parts are genomics-informed surveillance and policy, so looking at the policy surrounding pathogen genomics within the country, sample selection, collection, and referral, so that's just the laboratory part. Then we have laboratory workflow, so just processing the samples through the wet lab to collect, process, and sequence the samples. Then we have bioinformatics and analysis, so that's basically where you analyze the samples and generate some results. We then generate the reports, summarizing the results, and send it off to the end users. So that corresponds to phase 2 of the PG phase framework. And implementation and public health practice is just basically phase 3 of the PG phase framework, where end users aim to implement pathogen genomics results to inform their surveillance and outbreak responses against infectious disease and AMR. Within each section of the tool, we have up to 6 subsections per section. So those relate to the specific parts of each section of the tool. So in terms of surveillance and policy, for example, this could range from do you actually have a policy in place and do you have an implementation plan in place to do you have a financial plan in place to actually budget pathogen genomics. And within each subsection, there are items that people can respond to indicate the current maturity level across the subsection, and then that gets combined to provide a maturity level for the specific section. So this monitoring and evaluation tool can be used against a single system, and it can be applied both at a national level as well as a regional level. And those sections can be completed separately or together. And the sections can be split across a number of different departments within the public health laboratory and the public health department, so that we're reducing the participant burden. And as I've said, we've begun to pilot the tool in a few countries and using the pilot results to refine the tool. So what are the future directions in terms of monitoring and evaluation of pathogen genomic programs? So basically, we are wanting to incorporate monitoring and evaluation activities earlier in genomics implementation, and we're also measuring the quantitative impacts of pathogen genomics. So we're starting to actually do some studies on impact assessment on those. We're developing economic evaluation approaches, so we'll see a bit more of economic evaluations throughout the years. We're also evaluating training, mentoring, and capacity building programs, and I have heard that some countries are starting to want to implement training programs, so I feel it's important to actually evaluate those training programs to see how effective they are in upskilling the laboratory workers, and also evaluating metagenomics implementation as metagenomics is just being implemented into public health systems. So hopefully by the time we actually run program evaluation, we're hoping to use program evaluation results to implement pathogen genomics in more cost-effective ways, to increase the use of pathogen genomics in public health, particularly among end users. And lastly, to present a case for funding pathogen genomic services. And I think as funding gets increasingly competitive, I feel that it's important that we have the results to show that pathogen genomics is an effective tool for us to reduce the number of cases and the disease burden and the economic costs associated with infectious disease. And thank you very much.
Amazing work. Tons and tons of work there. Any questions for James? I know. Right over here.
Hi, this is excellent tool. I think there's a lot of interest in this. My question would be, once the tool is released, is the idea that institutions or laboratories, public health labs, health departments, would they complete this independently, or would it be like with assistance? And then how long, is there an idea from the pilots for how long it would take to complete each of these sections and get their maturity level?
Yeah, so just in terms of the first question, the tool can actually be done independently or with assistance, and ideally the tool would be the completion of the tool would be a collaboration between the public health unit and the public health laboratory, depending on which sections get completed. But ideally you want the two to work together so that they can share different perspectives in terms of the implementation of pathogen genomics within the country. And in terms of the piloting, we're currently piloting the tool in a number of countries, and within the pilot, countries are only completing a small number of sections, so they're not completing all of the tool. But nevertheless, it does take time for each section to be completed, and that is just based on collecting the information that is required to provide an accurate rating behind each subsection and the subsequent section that they're piloting.
I know everyone's probably starving, but are there any other final questions for James before we end this session? No? Oh, thanks so much, James, for that. Thank you so much to all our fabulous speakers. I know we learned a lot, and I have a feeling that you're probably going to get peppered with questions over the lunch break. So please join me in thanking everyone that has presented this morning one more time. It's now time for lunch. I think everybody knows where lunch will be served, and we will report back here at 2:15 for closing ceremonies. Thanks very much.
Hello. Hello. I love that your energy is the same now as it was on day one. All right, everyone, final session of GMI15. I am very, very pleased with, I think we've met a lot of the objectives that we came in to achieve. A lot of it is around connections. I saw everyone exchanging numbers during lunch, taking photos. That's exactly what we want this to be. So I'm very, very, very happy to be closing up GMI15. We've got 3 key people that we want to pull up on the stage. So I might hand over first to Adriano and Cesare for their host announcement of GMI16. Thank you.
Okay, I introduce myself. I'm Cesare Camma. I'm working in the same institute of Adriano in Italy. Our institute is called the IZS. It's a veterinary institute, but we have a lot of collaboration with public health labs. And okay, for the presentation of the next GMI meeting, our colleagues from Tunisia couldn't be here, but they prepare a video. But before, I would like to explain why from Italy we promote the next GMI in Tunisia. First of all, because we have a good collaboration with them, with the Ministry of Health and the main public health lab in Tunisia. And last year we started to work for a project that was funded by Italian government to improve capacity in pathogen genomics in Tunisia, and also to develop a platform for data sharing, genomic and metadata sharing in Tunisia from the different sector, from public health, veterinarians, and environmental. And the second point is that 3 months ago in Tunisia was organized a big conference on One Health involving ministries of the North African countries and the Middle East countries, Ministry of Health, and representatives of the different sector, health, veterinarian, and environmental. During this conference, it was established that Tunisia will be the hub for One Health, for promoting One Health in that region. So we think that Tunisia could be a good candidate to host the next GMI 16?
Yeah, the reason I'm here is just to say, if Torsten said that was a crazy idea to organize here in Australia, I think that this will be probably the craziest idea that we, as Italy, organize and help to organize in Tunisia. But you know, a bit of masochist. But I think that to have the first GMI in Africa, we are ready, and we will do most of our engagement. So that's it.
Okay. So start the video. The video can start.
مرسوم سواك بلوح بالي يا ليلي ليلي يا هليل ويا شباب
All right, I'm feeling pretty psyched for GMI16, I must say. Wow. Do you want to add something, Eric? I have no idea how they put that together. Please tell them how impressive that video was. Oh my goodness. Okay, and how beautiful that we get to bring GMI to so many corners of the world. I'm very psyched. I hope everyone that's here is also able to join us in Tunisia next year. All right, Next we're handing back to Masami, who's going to give us a wrap-up on GMI15 and perspectives from the GMI Steering Committee. So over to you.
Thank you very much. So, came to the end of the conference and with the exciting news from Tunisia. And then while that I'm very happy to have this proposal from different organizations and different countries, I think that GMI started from, of course, GMI 1, and then now that 15, and then 16 will be Tunisia, and then there will be more and more. So if you would like to host GMI in the future, you really should think about it. You may have some project and you may want to make it happen. Maybe, who knows, we might want to do it in Singapore or Bangladesh or Thailand, or, like all kind of different possibilities exist. So start thinking about it, keep communicating with the fellow here, and then so that it may come the possibility. Anyhow, the first thing that I wanted to say is that it has been so wonderful organization, wasn't it? So I want to give the round of applause to the organizers. Thank you so much. Flawless organization. Everything was wonderful, and we had such a great discussions, and then all the series of presentations. I was asked by a few people that the presentation files will be made available, or recording is made available, or yes, okay. So because I usually make these reflection slides and then always wonder why participants need reflection when everybody was here. Like, why do I have to summarize the things that you already saw? And then I noticed that these slides would be useful for people who want to summarize and report back in the country. So I just put together just for that purpose. So I'm going to skip very quickly through that. We had 3-day conference, and we had those many people here, and we had 5 different themes. And then the first theme was the data platform standards, and then theme 3 was the data sharing. That was such a rich discussions, it seemed. And then we had this enabling tools and infrastructures, and then we spoke up quite a bit about what could be the good practices from the different experiences. And then we talk about the policy and sustainability, and then hopefully that my summary is useful for everybody. And here, this is something that I put together from the morning sessions of yesterday and today, that we have had quite a few participants in the morning, and then we asked the question that what would be your wish list? What is the topics that you would like GMI to cover? So very interesting inputs. Individually, people told me that, we want to have AI to be a good topic to use as a together tool as WGS and then AI, or something about this mapping is necessary. There are lots of different initiatives, and then we may like to bring people here so that we can talk together. I think it is already happening. And other things like the water research, that was also like something people wanted to see. And then metagenomics, also the proficiency testing assistance to the low and middle income countries. There are quite a few items that we may like to think about. And thinking about those wish lists, the GMI Steering Committee will meet in either September or October online, and then you're more than welcome to nominate. If somebody wants to volunteer, just contact me. The idea is that current Steering Committee member can nominate anyone, and then it will be voted by the Steering Committee. So if you nominate somebody, I'm particularly thinking about having somebody from Australia. It would be really nice geographically, and then also thinking about the contribution from this year's GMI. So any other countries that if you think that you want to be more involved and then leading this initiative, please come to talk to many steering committee members here. And then three idea I got was that let's revitalize the working groups so that we can aim at making something very tangible and then practical output from GMI, not only just talking about it, but then maybe we can make some communication materials or some videos or something very useful for promoting this technology. And then also this inclusiveness, so we need to strategize that how we can make GMI inclusive. And then lastly, that there will be a very limited participation from the low and middle income countries because we don't have a sustainable funding. This is mainly voluntary participation. So steering committee should think about some sort of funding the strategy so that we can start including those participants and experts and researchers from the low and middle income countries. And then these are also I collected from various different people here in the room. The question was, what are you going to do after this meeting? So next Monday, you will be resuming your everyday work, and then you will do something about GMI, or you completely forget about GMI. So I want to ask you that please just to give few thoughts that what you can do to continue this effort, and a few things that the people wrote that they want to debrief their colleagues about the GMI meeting, that all the learning they want to share with the colleagues. That's a very good action. And then another thing is that some people didn't know so much about GMI. So now that Adriano has put together all the beautiful websites, so we have lots of information about GMI, so you can look up the website and then learn about the past GMI efforts, and then also the mission statement that has been renewed from the Canadian and then Barcelona meeting, so you can take a look at it and then learn more about it. And then also that this networking was a big deal. So I think that I talked to a few people that we exchanged the LinkedIn account. It may be a way to go. And then there are lots of people updating LinkedIn posts very much a regular basis. So I think that this way, if you contact me and then connect request is coming to my account, I would definitely approve everything. So I think that we can use those kind of social media tools to create a network and broaden the connection. And then lastly, this foster knowledge sharing. So we spoke a lot about data sharing, but then I think that the beauty of this GMI is that we will be able to meet in person and talk. And then the dialogue starter is the main aim of the GMI. So I think that this is something that would be useful for you. With that, this QR code is for the FAO informal network registration. So if you want to join the FAO network on whole genome sequencing, that you can register. And that's all from me. Thank you very much.
Right. Thank you so much, Masami. That was a very concise summary, and I'm sure everyone that is going to watch the recording is going to appreciate it very much. All right. Torsten and I are very happy to be closing now. So there's a couple of things we wanted to do first. Because not everybody was able to attend the earlier session, we'll do another Mentimeter so that we can get some broader thoughts. But if we get to the next slide, please. Yep. I'll hand over, before that, back one, sorry. I'm going to hand over to Torsten to give his impression of this GMI conference.
I'm doing that first.
Yeah,
Before
yeah,
I meant to,
yeah.
okay. When I opened this conference, I talked about how 11 years ago I went to my first GMI. Well, something else also happened 11 years ago. That was when I last organised a conference, and it was my first conference I organised, and it was a national conference for bioinformatics, and it then ended up forming a professional society which now has its own annual conference. And I swore to myself I would never do another conference again after that. I mean, it was hard, but you know, we had a great team behind us, and that's what made that conference a success. And the same thing has happened here. We had an amazing amount of support from the Institute, the organising committee, everybody, and we'll get to the details of that. But it was that support which made it happen. And I went to those GMIs before the pandemic. You know, I thought it was a good idea, but I felt it was lacking a bit of impetus. And then COVID happened, and then the reboot in Vancouver happened 2 years ago, and then Barcelona last year. And I felt that, you know, the need was even stronger now for a GMI type event and bringing people together. And that sort of made me and Tia think, well, this aligns with the Centre for Pathogen Genomics, is where we were working. We were working in the region. Really, GMI aligned with the principles and ideals and ideas that we had for the centre. And so, you know, we thought, I was thinking, I don't want to organise a conference, But I did go ahead. We did go ahead and organise a conference. And it was not me alone. It was Tiet and I. Tiet are sort of two sides of a coin. I guess I'm the more analytical, technical side. I don't really enjoy the governance, policy and treaty documents and that sort of stuff. I kind of want to get stuff done. But Tiet comes with that balance. Together we're sort of like the yin and yang in the centre. She has the policy, governance, legal interest, and she knows how to get people together from that side of the world, and I think I know how to get people together from the other side of the world. And together we thought we could do something good. And I think we did. I think we have achieved something good. I thought having a conference in Australia is usually, every conference, international conference that's held in Australia is usually has a 30% drop in attendance. That's kind of pretty typical, 30 to 40%, just because people can't afford to travel or they don't want to travel. But you guys, you surprised me. I think we outdid the previous GMI, both in numbers and in breadth of countries attending. And like, I just really thank you for making the effort to come here and be part of this GMI. I think it's been one of the best GMIs so far. Vancouver was good. But I think this was different. I think we just had a different type of people here. We're in a different stage now that IPSN and phage are moving along. And yeah, I think the best thing about GMI to me, some people may argue that it hasn't done anything or we're not sharing any more than we were, but I think we're establishing relationships and community. And those early, from that first GMI at York with Tim, I met people. That was where I met Marc. That's where I met Eric Brown. That's where I met that's where I met everybody. That's where I met the NCBI folk. You know, that one meeting, that kicked off a relationship that still extends today. Like, that's how things happen. You need these personal relationships. These random Zoom meetings are good, but once you get together and you kind of get to know the person and have a meal, sit next to them at a meal or drink with them, these are the relationships that are going to change the world for the better. I think you need personal relationships to make stuff happen, and I think that we've done a good job of increasing that and increasing our reach here, and I'm hoping that the next GMI and future GMIs will enhance that network and make it even better, and maybe we will really achieve our sort of utopian ideals about what we want in data sharing and a global kind of pathogen surveillance community. Thank you.
All right, so I'm sorry that we're following that up with a Menti exercise, but we have to do this. It's M&E. We all know how important M&E is. Could we please get the Menti QR code up? It's only 5 questions, so don't be too intimidated. But if everyone could please participate, this would really help with the next GMI and for us to improve, continually improve.
Yeah, this is not one of those surveys where they promise it'll only be 7 minutes, and you're there 30 minutes later still moving the slider bars.
All right, so we all logged in. 3, 2, 1. All right, can we please go to the web page for the Menti exercise, please? Okay, where have you traveled from? Locally, 20. Internationally, 32. Interstate, 5. So we have more international guests than we do local guests. That's a huge achievement for us. It was something that Torsten and I really, really wanted to reach. And I think that's very much represented in the opinions and the perspectives that have been shared over this week so far. That is awesome. Thank you. Let's go to the next slide.
Ooh.
Please place a pin of where you have travelled from. We need to screenshot this once everyone's put their pin down. Oh, wow. Okay, How good is your geography is the real question. If you don't know where your country is. Look at that. All right, Torsten and I are on a continents collecting exercise. We want to have as much representation as possible. Jess, can we somehow screenshot this once we... I don't know how to do that now. Oh my gosh, how cool is this? We weren't... Yes, South America is a big gap.
Several people live on sailboats.
I mean, can we say Josephina through you? Like,
I think this is Michael from Illuminati.
That's amazing. Thank you so much, everyone. That is such a fantastic spread. And now we know the next one we will have to definitely wheel someone out from South America. Wow, okay. Thank you so much. Let's go to the next one. What topics would you like to focus on? So Masami already raised a few that were brought up from this morning's session, but for those that weren't able to make it, what are the topics that you'd like to focus on or discuss at the next GMI? One Health. Oh, the nice. Starting small. Whoever put that in. AI ethics, next-seek giveaway. I'm just going to keep looking at Illumina every time I see a giveaway. I think we should do a competition at the next GMI. You win something. That would be a good one. Okay, sustainable funding, AI, One Health, drug targets, benefit sharing. Okay, fantastic. This is just a brain dump of ideas, so I'm going to let everybody continue to add things, and we'll do our best to consolidate and inform the next organisers. Microbiomes, that's something we haven't touched on yet. Okay, ice cream. Torsten dancing. Okay, it's becoming a joke. We're moving to the next one. All right, what was your most memorable part of this week?
It would have been Torsten dancing.
Lizards, snakes, networking. Okay, if the animals overpower the conference, this is going to be of concern. Data sharing views, meeting new people, the barbecue. I'm so glad that hit. First day talks. Yeah, lots of meeting people. Did I see honesty somewhere? Welcome to country. I'm really glad that that was received on everyone's end here. The breadth of discussion, international experiences. Okay, well, you guys have covered chicken the snake. Yep. 7 seeds. That's one of our cafes. I feel like Adrian wrote that. Did you write that? Okay. Fantastic. Thank you so much, everyone. It looks like networking is front and center. And to be honest, that was Torsten and I's main objective, was to get new people here and for them to network. That's awesome. Okay. Next one. I think it's the last one. And what can we improve on? Don't say we're perfect, because we are not. There are always things to improve on. Food. Oh, that's so personal.
Food.
Weather. More workshops. Okay.
I think that's fair.
Yeah.
Look, I got you 2 hours of sun yesterday, but I had to trade it for today. That was the cost.
More viral talks, more resources, bring back the breakout sessions. Yep, breakout groups. Okay, everyone's really wanting to get into some deep dive discussions. That's great. A conference report. Well, the conference isn't over yet, whoever wrote that. You may very well get that. Having a walkthrough data exercise on how it's gathered and used. A white paper. I mean, anything specific, please do let us know. Facilitated networking and icebreakers. Oh, interesting. I always try to hide from icebreakers, but I'm glad to hear that it's of interest.
I usually get forced to do the icebreakers at events, but that's
really good at them.
That is a good idea.
Yeah. Solutions from GMI15. Poster slot. Okay. I don't think we've done a poster slot before, Mark. Masami, have we ever? No. Yeah, we have. Yeah. Okay. Well, we should definitely.
Early GMI had posters. Yeah. York had posters.
Great. All right. Thank you so much, everyone. We'll definitely be gathering all of those thoughts. That's all for What Is It? I'm terrified. Giving Marc Emeritus. Okay, can we please get back to the slide deck?
What was it?
Coke Zero.
Yeah, I'm with
I think
that.
Torsten agrees.
I'm so over orange juice.
Okay, next slide, please. Over to you, Torsten, for acknowledgements.
Oh, sorry, what are we on?
The mic's right there as well.
Oh, this screen's different to that
Could
screen.
we change the facilitator screen as well, please? Thank you.
Obviously there's lots of people to thank. You can't make a conference like this without a lot of people, and I guess the first point of call is the organising committee for the conference. I really appreciate, because the time zone problem between Europe and North America and Asia, like, it does make it really tricky, but across the meetings we managed to get input from everybody, and we took your input, the steering committee's input, organising committee, seriously, and yeah, I think we factored in a lot. And I guess one of the key differences for this year was the ECR breakfast thing, which was something we haven't really seen at GMI. Like, all these ideas are great. How do you fit them into 3 days? You have to sort of pick and choose. So, yeah, thank you. Next. Oh, sorry. Sorry? The speakers and facilitators, of course. Obviously, this wouldn't happen without all the speakers, and I guess the speakers were sort of selected and invited to try and have a diverse geographic, gender, background, topic field, and I think all the talks were very good, and we put a lot of pressure on you. You know, these 10-minute slots flustered a few people, and I apologise a little bit for that, but I think it keeps the energy going in a conference. I think the 15 to 20 minutes, it gets a bit hard one after the other, but the 10 minutes keeps your message concise, and I really appreciate everybody keeping to time and following kind of instructions and not bringing their 500 slide deck to a 10-minute talk. And the facilitators during the workshop yesterday, we had lots of facilitators, and some of those people were volunteers. But we appreciate them stepping up and actually taking it seriously and going through Tim's details and following the steps. And I think that what's been gathered from that, I understand, will be a very, very useful data set for the IPSN. All right, I'll try and find the button. I haven't been good this meeting. But yeah, of course, none of this could have happened without some financial support. Obviously, GMI doesn't have any money in its coffers, so we did get the support from 3 great sponsors. So our primary sponsor was Illumina, who not just the team here, but Cara and others who initiated the sponsorship itself. But they were very open to discuss ideas and flexible. And without Illumina, this event would not have happened. That's 100% true. And like I said yesterday, they're a long-time partner of many of the researchers in the centre, and we hope to continue that relationship in the future. Our second sponsor was the City of Melbourne, which is great. You know, we love Melbourne. We think it's a great city to come to, and I think a lot of you recognise that as well, because you came here, you chose to come here. We also, I'd like to thank, and Councillor Andrew, who came and spoke about, he actually gave quite a good talk. I was surprised. All right, thanks to the Melbourne City Council. And lastly, the Centre for Pathogenomics. Not only did they contribute some money, that's our centre that many of our local here people are in, not only did they contribute some funds, but really it was the time they allowed their staff to spend on getting this event to happen, and that took a big chunk of effort. And thanks to my centre.
All right. Yeah. Oh, yeah.
You go. I think you should.
So Adrian and Masami and Ben, thank you so much for introducing a new session to GMI. I think that it was so very well received. We were so concerned. It's 8 a.m. Are you sure? And we ended up having twice as many people as you guys were wanting to reach. So you wanted 20. There was at least 40 every single day.
Yeah.
So
I was a bit concerned about it. 8 o'clock students. It's a bit early, but but when I turned up this morning it was actually, I thought, this is not just an ECR breakfast. This is like a GMI session. Like, the conversations that were happening this morning were could have been a GMI session, and that's the type of conversation that I love to see at these events.
So thank you so much.
Thank you very much.
Just a few more. The events team, who are all sitting at the back, they are unreal. So unreal. Thank you, Zach, who's been doing all of our IT work. Thank you to Kelly, Kimberly, and Jake, who are the ones that are managing this space. Our catering, the wildlife encounters or reptile encounters, the Woodward Centre, which is the Melbourne University Law Building, the Doherty Institute for supporting us and doing all the comms and advertising the networking sessions, all that kind of stuff. We couldn't have done it without you, so thank you, team.
Amazing.
Ben's going to hate this. Thank you, Ben, for your limitless support, for letting Torsten and I do whatever we want, for letting us go out on all these international endeavours and choosing who we want to work with and bringing it back and having trust in us that we're going to deliver and letting us be autonomous in all of that, and allowing us to really kind of design this the way that we thought it could work and hopefully filled a gap in the global space. So couldn't have done it without you. I know you hate this, but we wouldn't have been able to do it if you didn't support us.
Yeah.
And Jessica and Hassini, these two have done everything.
Yeah.
Hassini is unfortunately not here on our last day. She only worked 2 days a week, and those 2 days a week were all GMI. GMI breakfast, lunch, and dinner. She was incredible. She's been the one that's been emailing everybody, setting up all the talks, making sure that we ran smoothly, kept telling me that the animals were going to be just fine. And I'm like, are you sure? Are you sure they're going to be fine? She really brought this together up until this point, and then Jess delivered it all the way home. So thank you so much, Jess, and thank you, Hassini.
Yeah, sorry, I just have to second that.
Please.
I mean, if I had organised this conference, it wouldn't have happened, right? It's people like Jess and Hassini, and Chiet too. Chiet, she's being modest here, but she's behind the scenes as well, way more than me. Without them, it would not have happened. Just amazing logistical expertise and persistence and patience and focus. I envy those qualities. Thank you. I'm going to clap again, sorry.
All right, and lastly, all of you, thank you so much for being engaging participants. Thank you for embracing Australia, for embracing our traditional owners of the land, for embracing our food and our native animals. I really, truly hope that this has been a memorable experience for you, and I hope that it encourages you to come back, to collaborate with Australians, to connect with our students, all of the above. This wouldn't have happened if you guys didn't ask questions, get engaged in the conversations. It's a meeting, not a conference. So we wanted people to discuss, and we hope that you got as much out of it as we did putting into it. So thank you very much.
That's you guys. With that, I call the conference closed, and I'll see you in Tunisia. And I hope everyone has safe travels home. Thank you very much for coming.